So in this video, I'm going to cover what is going to be on your final. And I'm going to start with the cumulative final list. So these are the activities that will be from Lab Practical Exam 1 or Lab Practical Exam 2. And so I will review each of these activities. And so this will include the gram stain, the endospore stain, the acid fast stain, capsule stain, OF glucose, phenol red broth, IMViC, decarboxylase, deaminase, and then lastly with the Kirby-Bauer. After I review these, then I will review the new material. You want to know this slide. This is going to be important. You're going to see this in lecture and lab. And so you want to understand the different types of morphology. So that spherical is a coccus, a rod is a bacillus, a curved rod, what looks like a comma, is going to be a vibrio. You want to know that a spirillum is a rigid spiral and a spirochete is a flexible spiral. Those are morphology. Those are shapes. You should also know the arrangements. So diplo, di means two. It's two of those cells together. Staphylo refers to grape-like clusters. And strepto refers to chains. Now, staphylo can be Staphylococcus, but there isn't Staphylobacillus. And that has to do with the way that bacillus divide. They will divide along the horizontal plane, meaning they'll divide out this way. So bacillus can form streptobacillus. They can form chains because they can be, in a row, but they're not going to be in clusters because they don't divide vertically. They only divide horizontally. So you want to know this slide in terms of the names for the different types of morphologies and the names for the different types of the arrangements as well. Now moving on to our capsule stain. Again, the capsule stain is going to take the place of the standard plate count. So this one is in place of the standard plate count. So you want to know for the capsule stain, what is the purpose? To differentiate between capsule producing cells and unencapsulated cells. So meaning that this is a differential stain. You want to know the functions of the capsule. So all of these functions. You want to know the composition. So what is the capsule itself actually made of? So that mucoid polysaccharide, which is a carbohydrate or a sugar, or polypeptide, protein-based. It's less common, but is used in Bacillus anthracis. You, again, will see this also in lecture. For the capsule stain, you should be able to tell me that the cell is stained with safranin. The safranin is going to be our basic stain. It stains the cell. You want to be able to tell me that the background is stained, and that's because we use an acidic or a negative stain, like Congo red, and that the capsule is the clear area. So those clear areas around the cell is the capsule. So you want to be able to know the different parts. The capsule is clear, the cell is red because of the safranin, the background is red because of the Congo red, and so understand that amount of detail in terms of the staining procedure. In the capsule stain, you don't have to memorize the procedure. However, you should know which dyes are used and what part that stains. So again, the safranin is used to stain the cell, and that's a basic stain, and that the Congo red, which is an acidic or a negative stain, is used to stain the background, while the capsule is the clear part. So again, you don't have to memorize this procedure. You just need to know, like I told you in the slide before this. Same thing with this. You don't have to memorize the procedure. You just need to know that we use two stains, that we don't use a heat fix step. And so you just want to know what the capsule stain will look like. You should be able to identify, not necessarily for the quiz, but for the exam, you should be able to identify that these are examples of a capsule stain. You don't have to distinguish between if you're looking at Klebsiella pneumoniae or if you're looking at Bacillus megaterium. You don't have to make that distinction of which is which, but you should recognize that if you see either of those pictures, that in fact you're looking at a capsule stain. Because again, the background is stained with the Congo red, the cell is stained with the safranin, And that clear halo around the cell is going to be the capsule. And so when you're studying for the practicum, you will need to know an example organism. And I believe the one that's on our list is Klebsiella pneumoniae, which causes pneumonia. And then lastly, we have our gram stain. You want to know the purpose of the gram stain. So to stain and differentiate gram-positive versus gram-negative bacteria. You want to know why it's important to do a gram stain. And so in that video, I talked about why it's important to do a gram stain. So to pick the appropriate antibiotic, as well as if you have a gram-negative infection, I talked about why you would choose a particular drug or not. So go back and review what is the reason. So it's not written down, but I did talk about it. What is the reason that we need to know if an infection is caused by a gram-negative or a gram-positive bacteria? And there were two main reasons. So you'll want to go back and review that. You want to know that in the gram stain that we use basic dyes to stain the cell. You want to know that we use two dyes. We use crystal violet and we use safranin. You want to understand that at the end of this procedure, gram-positive are going to appear purple and gram-negative are going to appear pinkish red. And so this would be like an example of what you would see when you guys did this, because you took your Citrobacter fundii, which is your gram-negative, and you mix it with Staphylococcus epidermidis. And I told you guys that you would mix it in the same puddle of water. And so in that puddle of water, you're going to have both your gram-positive and your gram-negative. You want to know why gram-positive stain differently than gram-negative. And that is that the gram-positive will retain the purple color. They will retain the crystal violet because it has a thick layer of peptidoglycan. So because of this thick layer of peptidoglycan, it's going to hold in that purple crystal violet. Whereas for gram-negative, you want to know that the gram-negative has an outer membrane, that it has these LPS, this lipopolysaccharide, which has lipid A, which is an endotoxin. You want to know that gram-negative have porins in their outer membrane. And then again, the big key is going to be that they have a thin layer of peptidoglycan. So that when we decolorize with the acid alcohol solution, the acetone alcohol, when we decolorize, because of the thin peptidoglycan in gram-negative, you want to understand that the purple dye is going to come out, that the crystal violet is going to come out, which is why we have to stain with the counter stain, which is going to be safranin. And so the reason that this stain is differential, it differentiates based on the thickness of the peptidoglycan. Gram-positive have a thick peptidoglycan. They will retain their crystal violet. Gram-negative has a thin peptidoglycan. They will decolorize and they will stain with the secondary stain, which is the safranin, in this experiment, and they would appear pink-red. You want to know the overall steps in terms of how to make a heat fix smear. You want to know that when we transfer bacteria to the slide, that we want to use a needle. This is a key, a needle, because we don't want to pick up too much bacteria. You want to know the purpose of the heat fix, and that's on the next slide. So why do we heat fix? You want to know the three reasons, one of which is the mechanism, meaning the way that this works. So we heat fix to kill the cells, to adhere bacteria to the slide, and the mechanism, and the other reason we heat fix is that proteins denature and coagulate. So again, that means that the proteins unfold and they stick together. And so that's going to kill the cells and adhere bacteria to the slides. This is one of the few procedures where you do need to know the steps. You do need to know the steps for this one. So you should know that the primary stain is going to be the crystal violet that we add for 1 minute. And then we dump and we rinse with deionized water. You want to know that we add the mordant. In this case, it would be the iodine. We add the iodine to the slide, allow it to sit for 1 minute, dump, and then rinse with deionized water. You want to know that for the decolorization step, that you're going to use an acetone alcohol solution until the solution coming off runs clear. So again, that step is not 1 minute. It's simply until the liquid coming off is going to be clear. You want to know that the decolorization step is the most important step in the entire procedure. That this is the part where the stain becomes differential, meaning that this is where gram-positive and gram-negative become different, is during the decolorization step. You want to know that then you rinse with water and add the secondary stain, or what we also call the counter stain, which is going to be our safranin. We add it for 1 minute, then dump off the safranin and rinse. And then we would view our slides under the microscope. You want to know that the crystal violet is the primary stain. You want to know that the iodine is the mordant. And you want to know the purpose of the iodine. And the purpose of the iodine is that it forms a crystal violet iodine complex, basically meaning that it makes the dye molecules larger so that the crystal violet is retained within the cell. So you want to understand that when we add crystal violet, that both gram-positive and gram-negative are purple because they both stain with that basic stain. When we add the iodine, which is the mordant, which helps keep the crystal violet in, both cell types are still purple, right? Because they still have their crystal violet. But the most important step of the entire procedure is going to be the acetone alcohol. And this is where the stain becomes differential, because gram-positive have a thick layer of peptidoglycan. They will retain the crystal violet. Gram-negative have a thin layer of peptidoglycan, and so the crystal violet is going to come out of the cell. And then you want to know that the last step is the safranin, which we call either the counter stain or the secondary stain. It's the second stain that we use. It is still a basic stain, so it still stains the cell. In the case of the gram-positive, the safranin still gets in, so it's not that there's no red in the cell. It's just that we don't see it because the purple is darker than the pink. So at the end of the procedure, gram-positive would appear purple. Gram-negative would appear pink-red because they became colorless during the decolorization step. But this is why we need the secondary stain, because we need to be able to visualize those gram-negative bacteria. So this is why we use our secondary stain, which is going to be our safranin. So you want to know for each of these steps what color gram-positive appear, what color gram-negative appear, and why. So you should know those different steps. You should know what each of those solutions is used for. So again, the crystal violet is the primary stain, the iodine is the mordant, the acetone alcohol is the decolorizer, and the safranin is going to be your secondary or your counter stain. The other thing that you want to review, and I mentioned this in the video, is I want you to review what does it mean when we say that we over decolorize? What would the cells look like if I over decolorize? And then on the flip side, what would the cells look like if I under decolorize? Meaning that I don't run the decolorizer long enough. So you should be able to tell me why either of those are not good, why you don't want to over decolorize or under decolorize, and how that affects the results. So these are the things that you want to know for your gram stain. This is what I was talking about, how you need an example organism. So if you were given an image that looks like this, where you can see little pink rods, you know that an example organism would be E. coli or Escherichia coli. So E. coli would be an example of a gram-negative rod. Notice that it's pink because it stains with the secondary stain. If you see these little spheres of clusters, right? So these grape-like clusters of little spheres, and they're purple, you're looking at Staphylococcus. And an example of this, Staphylococcus aureus, Staphylococcus epidermidis, those would all be examples. So use the one that's on that first slide when you're studying. But what you should know is you should be able to look at these pictures and say whether it's gram-negative or gram-positive, and then also an example organism that would be possibly on the slide. And then lastly, you want to review this slide. So this was a question that's asked at the end of the video. What would each step look like if we accidentally added the safranin first and the crystal violet second? So you would want to be able to kind of describe what colors they would be after each step. And again, we will go over this in our Zoom meeting to talk about the answers, but you're going to want to study this once we go over it. There's a reason that we have to add crystal violet first. So you're going to review this slide once you get your answer. And so this concludes our video on what to study for quiz 1. And so for the endospore stain, you want to know the purpose, and the purpose is to stain and visualize endospores and differentiate spore-forming cells from non-spore-forming cells, and this is a differential stain. So you want to know the purpose. You want to understand when endospores form, and endospores form when conditions become harsh. And so you want to know these different conditions for when bacteria would experience their condition as being harsh. So when temperature changes, or pH, or dehydration, or UV, or nutrient loss, or changes in oxygen concentrations, that is a condition that bacteria would experience as being harsh. And when they basically experience these harsh conditions, they will go through the process of sporulation, meaning that they will form an endospore. You want to know that the endospore is made of the protein keratin, which is found in skin. And so because of that tough keratin protein, it makes the endospore very resistant to these differences in changes. They're very heat resistant, resistant to chemicals, etc. So you want to know that endospores, what they're made of, what types of conditions would cause bacteria to form endospores. You want to know that eventually the vegetative mother cell will break down. It will give rise to a spore. And then when conditions become favorable, the bacteria will undergo germination and will go back to being a vegetative cell. And this cell is the one that's metabolically active. It's the one that's actively dividing. So you want to know the purpose of the endospore. What is the name of the process when they form the endospore? What is the name of the process when they go back to being a vegetative cell? What is the endospore made of? What are the harsh conditions, etc. In terms of examples of endospore producing bacteria, again, you don't need to know the whole list. I gave you the organism that you would need to know, and that is that Bacillus anthracis would be your example organism of a bacteria that produces an endospore, and that causes anthrax. You also want to know that there are 2 genus of bacteria that are most commonly going to have endospores, and that is Bacillus, which is aerobic. So you want to know this, Bacillus that's aerobic, and Clostridium, which is anaerobic, and that both of these genus are gram-positive bacteria. So more commonly, endospore-producing bacteria will be gram-positive. There are some exceptions, but generally speaking, endospore-producing bacteria are the ones that are gram-positive, and usually Bacillus or Clostridium. Now, in terms of the staining procedure of the endospore, you don't need to memorize the steps. However, there are some keys that you need to know, and that is that you have to apply steam when the malachite green is added. And that's because you need that steam to get the dye into that tough keratin protein. And so we steam our slide to get the dye to go into the endospore. So that you should know. You should know this diagram. Basically, the general understanding of when we apply the malachite green, that both endospores and vegetative cells will both stain green. When we decolorize, you don't have to remember that you decolorize with water, but you should know that if you decolorize, that the endospore will stay green, The vegetative cell will become colorless, meaning that the dye will come out. And then you should know that we use safranin as a counterstain. Again, you don't have to memorize the time or anything like that, just that you use safranin as a basic stain, and we don't apply heat, and the safranin is going to stain the vegetative cells, so that when you're finished, you end up with red, pink vegetative cells and green endospores. So again, you don't need to know the specifics of the procedure, but you want to understand an overview and how that affects the colors at the end of this experiment. You should be able to recognize a picture as being an endospore stain. You don't have to memorize that this was Bacillus polymixa. You're going to use the example organism of Bacillus anthracis. That's the one you want to study. So you don't need to know which organism produced the slide. You should just know that this would be an example of an endospore stain because we have that teal green endospore and we have our red pink vegetative cells. And so this would be what an endospore stain would look like. Now, moving on to our acid-fast stain. You want to know the purpose, right? And the purpose is to stain acid-fast bacterial cell walls and differentiate acid-fast positive bacteria from non-acid-fast bacteria. Again, this is a differential stain. You should know that organisms within Mycobacterium would be acid-fast positive and that they have a cell wall that is made of 60% mycolic acid. And then you want to study what are the purpose of the 60% mycolic acid. What advantage does it give bacteria by having this mycolic acid in their cell wall? And one, it makes the bacteria sticky, so it's an adherence factor. It allows bacteria to adhere to surfaces. It's sticky and it's waxy, which protects from phagocytic digestion, so meaning that the immune system doesn't break down that bacteria, protects the bacteria from dehydration. So again, bacteria can survive longer on surfaces as a result of having this mycolic acid. It makes bacteria resistant to chemicals, both antibiotics and disinfectants. So you want to know what is the acid-fast cell wall made of and what is the purpose of that acid-fast cell wall. I won't ask you to differentiate between the Ziehl-Neelsen method and the Quignon method. This was just more kind of for your information, and so you don't need to study the differences between these. In terms of the procedure, again, you do not need to memorize the procedure for this. You should, however, understand that we have to steam with carbofuscin. And the reason that we have to steam with carbofuscin is we need that carbofuscin to be able to penetrate that waxy mycolic acid. And so if we don't steam with carbofuscin, acid-fast positive bacteria will not stain because you need that steam to get the dye in. So you should know that we use carbofuscin with steam. You don't need to know which decolorizer and for how long for the acid-fast stain. You should know we decolorize. And then you want to know that there's a counter stain and that the counter stain is the methylene blue. Again, you don't have to memorize the procedure, but just know that we add methylene blue so that we can stain non-acid-fast bacteria. And the next slide will show you why. So again, this is just an overview of the staining procedure. You don't need to memorize this diagram. You should, however, be able to tell me what acid-fast positive bacteria and acid-fast negative bacteria would look like if given these different steps, right? So that initially when we add the carbofuscin, both cell types are going to stain pink because the carbofuscin is going to get into the cell. Then we decolorize, and when we decolorize, acid-fast positive will retain the carbofuscin. Acid-fast negative will lose their stain and they will become colorless at this point, which is why we need our methylene blue, because the methylene blue is our counter stain and it's used to stain the acid-fast negative, We do not apply heat when we add our methylene blue. And so the methylene blue will only get into the acid-fast negative and not the acid-fast positive. And so you should know at the end of this procedure, acid-fast positive bacteria will be pink. They're kind of a magenta, like bright pink color, where Staph epidermidis would be blue, or the acid-fast negative. You don't have to memorize that example, but that the acid-fast negative would be blue because it would lose its carbofustion in the decolorizer step. So again, in terms of what organisms you need to know for this, you don't need to know Mycobacterium leprae or Mycobacterium smegmatis, but you should know an example organism for an acid-fast stain would be Mycobacterium tuberculosis, which causes lung TB. You should be able to recognize an acid-fast stain. So you would want to recognize that this is an acid-fast stain. Notice we have our blue cells and then our magenta cells. The blue cells are the acid-fast negative. The pinkish color cells are going to be the acid-fast positive cells. Now, sometimes students panic because they think that this picture looks similar to the gram stain, and you might be right. So oftentimes, if I give you this picture, I might give you some other detail that would help you determine that it is an acid-fast stain and not maybe an not a gram stain. For example, I could say something about, you obtain a sample from a sputum sample, which remember is that coughing to get the fluid from the lungs. And that sputum sample might indicate to you that it's likely going to be an acid-fast stain. So there would be some key that might help you differentiate between the acid-fast stain versus the gram stain, because they do look somewhat similar. Not quite, because these are blue and the gram stain, they're purple. And then the pink is very different. In the acid-fast stain, it's kind of magenta. It's a bright pink, which looks different than in our gram stain where it's like a light pink. So just be aware, you should still be able to recognize this would be an acid-fast stain picture. So now we're moving on to Lab Module 2. And Lab Module 2 is dealing with our biochemical tests. So one of the things that you're really going to see throughout this part is that you are going to need to study those biochemical sheets. And those biochemical sheets are something we're going to review, but basically you need to know the substrates, the enzymes, the products, if a pH indicator is used, if a reagent is used, what does a positive test look like, what does a negative test look like, etc. So for the phenol red test, you should know that the purpose of this test is to determine if bacteria can metabolize a specific carbohydrate. And that carbohydrate can change. We can use pretty much any carbohydrate. We could have lactose in the media, we could have glucose in the media, any particular carbohydrate. You don't have to know the 4 bacteria that we used in this experiment. That is not important. So for this slide, in terms of what you need to know for this, you don't really need to know anything for this, except that the purpose of the Durham tube, that tube that's inside one of the tubes, that glass tube inside there, is used to detect the production of gas. Because some bacteria, when they utilize sugar, they will produce gas, and the Durham tube will allow us to visualize if gas is produced. You don't have to memorize that the plastic cap was the glucose and the metal cap was the lactose. That's not important either. So for day 2 of our phenol red broth, now when you're studying, students kind of panic about this in terms of, do I need to memorize all of the ingredients in every recipe? And the answer is no. So you do want to understand that in the phenol red broth test, that there are peptones, and that in that media, the peptones are there as food for general growth. Basically, it's providing these chemicals, these nutrients, like a source of nitrogen, for example, that bacteria would need to grow. And as a result, in a phenol red broth test, you should see growth to say whether it's a positive or a negative. If you get no growth, we say that that is inconclusive. And I'll come back to that in a little bit. You should have a general understanding that sodium chloride is there for salt balance because, again, you don't need to memorize it for this recipe, but in general, that the reason sodium chloride would be in media is for salt balance because, again, you're going to see that over and over again. You should know that the media contains a carbohydrate because that is our substrate in our experiment, meaning that's what we're testing if bacteria can metabolize. And our carbohydrate is only going to have one specific sugar per tube. So in our experiment, we have one tube that has glucose, and we have another tube that has lactose. You want to understand that in this test, we use phenol red, and phenol red would be put down as your pH indicator. And then you want to know what the different colors tell you so that you know that yellow is acidic. Remember that for everything except methyl red, every other pH indicator we use, yellow is acidic. Red, right? So like a pinkish red magenta color would be on the alkaline side. So this would be our uninoculated tube. It's kind of this, this is an orangey red. So that's why it says red orange. That's the neutral. Whereas the pink, so kind of a magenta color, is going to be the alkaline. So this is used to help you fill out your biochemical sheet. So again, you want to study your substrate, your enzyme. In this case, it's various endoenzymes. We're not testing any one particular enzyme. You want to understand what the products are. So the products of this process are acids, alcohol, plus or minus gases. So those are your products that you're going to record. You want to record what does the positive test look like, right? So the positive test would be yellow with turbidity. You want to record and understand what does the negative look like, and that is that red pink with turbidity. So the bacteria grew, but they did not use the sugar. Instead, they produced alkaline products, which tells us that the bacteria used peptones as food. You need to know that no growth, if you see no growth in this test, no growth, the interpretation would be no result or inconclusive. We can't say if the bacteria can metabolize the sugar because it didn't grow. And we can make that assumption because this is complex or undefined media where there are multiple food sources. Bacteria should have been able to use something to grow. And so this slide is really important in terms of your understanding. You want to understand why your readout needs to be done between 24 and 48 hours. So that's an important part. And so the reason that your readout has to be done between 24 and 48 hours is because when bacteria are given the choice between metabolizing proteins or sugars, if they can metabolize the sugar is going to be their preferred food source. That's what they're going to use first for food. As they do that, they will start to lower the pH, and the pH will go down, and that's why our yellow is going to be acidic, and that's going to be our positive. But once we get to about 48 hours, the sugar is going to begin to run out because as the bacteria are growing, they're utilizing that sugar as a food source. That sugar is going to run out, and then bacteria have no choice but to then use the peptones as a food source. And remember that when bacteria use peptones as a food source, it's going to produce alkaline products, and the pH is going to go back up, and the tube is going to go back towards being orange, or if long enough, it could turn magenta. And so if you read this out at, let's say, 96 hours, you would see a tube that's pink, and you might say, well, that's a negative for the metabolism of the sugar. Well, no, not really. It's just you waited too long, and the bacteria then metabolized the proteins, which is what caused what we call a pH reversion, meaning the pH went back the other direction as the bacteria started to metabolize that protein when the sugar ran out. So you want to understand the importance of why you need to do this readout between 24 and 48 hours. You do not need to memorize the results of this experiment. I will not ask you, what was E. coli's reaction or what was Proteus vulgaris's reaction. That I don't care. What I do want you to take away from this, though, is if you were shown a tube like this, you should be able to tell me what that tells you, meaning what is your interpretation. So if we see yellow with turbidity, right? Yellow with turbidity is going to indicate that the bacteria grew, right? The yellow tells us that it is acidic. And if we look in the Durham tube, we can see the gas in the Durham tube. So acidic and gas is going to be positive for the metabolism of glucose because it produced the acidic products, which then tells us that it metabolized the sugar. If you see tubes that are pink, so we see this turbidity and it's pink, that tells you that this tube is going to be alkaline. That's the K. It's alkaline, which is a negative for sugar metabolism. Because if we see alkaline products, that means that the sugar was not metabolized, but instead the peptones or the proteins were used. And that produced the alkaline products, which raised the pH. You want to understand what it means to say that bacteria are non-saccharolytic. And non-saccharolytic means that they don't use sugar as a food source. So Alkaligenes faecalis would be a bacterium that is non-saccharolytic. It doesn't use sugar. Instead, it's going to use protein as its food source. So again, you do not need to memorize this image. You do, however, want to be able to interpret. If you were shown this, you should be able to tell me what your interpretation would be. So what is the pH like? What does that tell you that the bacteria use for food, etc.? So this is basically your biochemical sheet. And so for each and every biochemical test that we do, so phenol red broth, our OF test, our indole test, our methyl red, Voges-Proskauer, citrate, etc., you want to make these biochemical sheets for that. And so for each of those tests, you should know the name of the test, the name of the medium, the substrate, the enzyme, the products. What does a positive look like and why? What does a negative look like and why? Is there a reagent in that experiment? Is there a pH indicator in that experiment? And if so, what is it and what does it detect? And then additional notes. This would be where you would put other things that were talked about in the slides. So for example, in the phenol red broth, under additional notes, I would include the purpose, right? What's the purpose of that test? What does it mean to say that bacteria are non-saccharolytic? Basically, you want to understand your interpretation or your readout. Why do you need to do the readout between 24 to 48 hours? I would put that in the additional notes as well. So now we're going to move on to our oxidation fermentation or OF test. So for this test, you want to know the purpose. And the purpose of this test is to determine whether bacteria can oxidize and or ferment glucose. So not only are we looking at do they metabolize glucose, but do they do it in the presence of oxygen, which is oxidize, or do they do it in the absence of oxygen, which would be our fermentation? So for this, you want to understand that these are agar deeps. So they are a semi-solid. And so when I inoculated this, I had to stab inoculate with a needle. You want to understand what was the purpose of putting mineral oil on one of the tubes. And the purpose of that is that the mineral oil is used to seal out oxygen. This is your anaerobic tube, meaning your tube without oxygen. And this one is testing for fermentation, while the tube without the mineral oil is testing for oxidation. And so that's what you want to take away from this slide. So when we look at the readout, Again, we have our recipe here. You don't have to memorize every ingredient that's in there. Again, you should understand a general idea that the purpose of a buffer is to keep the pH constant. You want to know that the agar is there as a thickening agent. Again, it has a low concentration. It's a semi-solid. And the purpose of that is that the agar is going to help to exclude the oxygen for the fermentation tube. You want to know what pH indicator was used in this test. So our pH indicator in this test is your bromothymol blue. Again, the blue tells you the alkaline color. So blue is alkaline. Yellow is acidic. Again, for every pH indicator we use except methyl red, yellow is acidic. So yellow is acidic. And then halfway between blue and yellow is going to be green. Green is neutral. And you want to know that our test has glucose because glucose is our substrate for oxidation or fermentation. So for this test, you want to know, again, your biochemical sheet. And so your substrate is going to be your glucose. Your enzyme are going to be various endoenzymes. So again, it's not just testing one enzyme. You want to know that the aerobic tube, the one that has no mineral oil, the one that's testing oxidation, meaning oxygen is present, that product is going to be acids. However, when we are looking at the anaerobic tube, the one that's sealed with mineral oil, that is testing fermentation. And when fermentation occurs, the products would be acids, alcohol, and plus or minus gases. So you want to understand the difference between the oxidation and the fermentation. You do not need to memorize which organism gave you which result. Again, the importance though is that you understand the readout. So if given tubes, you should be able to tell me what that tells you. So for example, in this one, yellow on the top, green on the bottom, and green throughout on the one with mineral oil tells me that it is positive for oxidation, negative for fermentation. And so you should know this term, which is that if you see an organism like this, that it would be a strict oxidizer of glucose. If you see yellow, yellow throughout, so both tubes are going to be yellow, yellow. Again, you don't need to know which organism, but you do need to know that that means it's positive for oxidation. It is positive for fermentation. So this organism would be an oxidizer and a fermenter of glucose. And then you want to know that that's referred to as a facultative anaerobe. And a facultative anaerobe will grow with or without oxygen, but grow better with it. And then lastly, we have our last set of results where we see blue on the top here, green on the bottom. This one is green throughout. So for oxidation, it's negative for oxidation. It's negative for fermentation because neither tube is yellow. So that organism would be a non-oxidizer and non-fermenter of glucose. And again, that term would be non-saccharolytic. They do not use the sugar, but they did use the proteins aerobically because the top is blue, meaning the pH is alkaline. And so that alkaline pH means that the organism used the peptones. And so you want to understand the readout, but again, you don't need to memorize which bacteria gave which result. I won't ask you that question. So this is just showing you bigger images of those results so that you could see them a little more clearly. So again, you can see that the E. coli is yellow and yellow. You can see that the Pseudomonas aeruginosa is yellow on the top. So again, positive for oxidation, but negative for fermentation. Alkalogenes faecalis is negative for both, but we do see the blue on the top, which means that they use the peptones aerobically. And so the last part of this is going to be looking at our IMViC. And so for your IMViC, you don't need to memorize which bacteria were used in this experiment, but you should know that there are 3 tubes that need to be inoculated. We have our MRVP broth, we have what's called our tryptone broth, which you'll see in another slide, and we have our citrate slant. You want to know for the citrate slant that you use a needle to inoculate it. You do zigzag inoculate the slant, but you need to use a needle for a citrate slant because you don't want to transfer too much bacteria. That will affect your interpretation. So now let's look at the readout. So you should know that the IMViC is the second most important ID procedure after the gram stain. So again, this plays a very important role in identifying which bacteria is present. You should know what each of the letters in IMViC stands for. So that the I stands for the indole, the M stands for the methyl red, the V stands for Voges Proskauer, and the C stands for citrate. So you should know what those different letters stand for. Again, you should know the name of the media. Those will be used on your biochemical sheet. So for both the MR and the VP biochemical sheet, the name of the media would be the MRVP broth. For the indole test, the name of the media would be recorded as being a tryptone broth. That's the name of the media. And for the citrate slant, the name of the media is a citrate slant, or more specifically, you'll see it's called a Simmons citrate agar slant, but citrate slant would work as well. So for this experiment, you would want to understand the readouts. This slide is a summary, but I will go into this in more detail for each one of these tests. So you don't have to worry about studying necessarily this slide specifically. So for your indole test, you want to know the purpose, right? The purpose is to determine whether bacteria can produce indole from tryptophan, and tryptophan is an amino acid. So you want to fill out your biochemical sheet, and that is that your substrate is going to be your tryptophan, and tryptophan again is an amino acid. This is my substrate. The enzyme that would need to be recorded is tryptophanase, which is an endoenzyme. It's an enzyme that's going to stay within the cell. For the products, you're going to record that there are 3 products. We have indole, we have ammonia, and we have our pyruvate. You want to know that this test cannot use a pH indicator. You can't use a pH indicator because the pyruvate is an acid and the ammonia is a base, and they cancel each other out, and you won't be able to detect a change in the pH. So for this test under pH indicator, you're going to put none and then describe why. For reagent, you're going to record the reagent is going to be your Covax, and you want to record that it detects the presence of indole. So that's what this reagent will react with. Covax alone is a pale yellow color. So the negative for this test is that the tube stays yellow, which means that no indole was produced, and bacteria do not produce tryptophanase because no indole was produced. The positive is going to be that pink red, and if we see a pink red, it tells us that indole is present. So the Covax reacted with the indole, and it turned that pink red, which is our positive. So this is basically your, again, biochemical sheet. You don't need to memorize which organism did which. So you don't need to memorize that E. coli was the positive and Enterobacter is the negative. But you want to know if you were given this result and you were told that this was an indole test, or you were told that this was a tryptone broth, then you would know it's the indole test, and you would know that this one with the pink red is the positive, and the one with the yellow is going to be the negative. So for your methyl red and your Voges Proskauer, so you have 2 separate biochemical sheets for this. They do have the same purpose. So the purpose is to determine whether bacteria can ferment glucose into mixed acids or acidoin. So we're looking at what type of metabolism do they do. In this test, our glucose is our substrate. So our enzyme is going to be various endoenzymes. And then for the methyl red sheet, the products are going to be our stable mixed acids. These are your products, and those stable mixed acids will decrease the pH. On the biochemical sheet for the methyl red, you're going to record the pH indicator and the reagent as being methyl red. In this case, methyl red acts as both because it's added after we do the incubation step, and it also changes color depending on the pH. So you would want to record that the pH indicator and the reagent is methyl red, and that methyl red is going to change color if the pH decreases. So it will turn pink red if the pH decreases. For the VP test, you want to record that at 24 hours, acids would be produced. So this is why we can't do our readout for VP at 24 hours. We have to wait the 48 hours. And so at the 48-hour time point, the bacteria will take those acids and they will convert them into a product called acetoin. And that product is what is going to be detected in the VP test. And acetoin is going to be a base. It will increase the pH. And so we add the VP reagents. It's called VPA and VPB. And those reagents will change color, meaning they will turn pink red if acetoin is present. So they will detect the presence of the acetoin. They're not a pH indicator. They are a reagent. It's the VPA reagent and VPB reagent. Again, for the VP test, there is no pH indicator. So for this slide, again, you want to understand that you cannot do your readout at 24 hours because for VP positive organisms, they will produce acids initially. So you want to understand that for MR positive organisms, that they will produce stable mixed acids, which will keep the pH low. For the VP positive organisms, that they will produce acids initially, which lowers the pH, but then will convert to acetoin, which will then raise the pH, and then the VP reagents can detect the presence of the acetoin. And so you want to understand question number 90 that was in the question set. So why are organisms that are MR positive usually VP negative and VP positive usually MR negative? And so I explained in that if an organism is VP positive, then it means that they produce acetoin, right? Because VP reagents will detect acetoin. So if they are VP positive, then they produce acetoin. Acetoin would raise the pH of the solution and therefore would not test positive with methyl red because methyl red only changes color if the pH goes below 4.4. So that's why organisms are usually one or the other, but not both. So for this particular slide, what you want to understand is that you inoculated the MRVP broth and then you allowed it to grow for 48 hours. And then you had to transfer the broth into 2 tubes. One tube got the methyl red reagent, one tube got the VP reagents. You do not need to memorize all of these acids and all of these products that can be produced for MR positive. You just want to know that for MR positive, they produce stable mixed acids. So MR positive, right, your positive result in this case would be if it's pink red. And if it's pink red, that tells us that acids were produced and the pH was less than 4.4. So that would be MR positive. MR negative would be not pink red or yellow or broth color or whatever other color you want to call it, but not pink red is the key. And so if it is not pink red, that means that acids were not produced and that the pH was greater than 4.4, meaning the pH did not go down for the methyl red to detect the acids. So that would be MR negative. So you want to know what the positive looks like and why. What does the negative look like and why. For your VP test, you don't need to, again, memorize all of these details. I'm not concerned with that. What I want you to take home from the VP test is that we will be detecting the presence of the acidoin. If acidoin is present, the tube will turn pink red. There are two reagents, VPA and VPB. And again, those will change color with the acidoin. And so VP positive would have pink red. And what that tells us is that acidoin was detected. Acidoin is present, and so the VP caused the tube to turn red in color. If the tube is not red, so if it's a coppery color or what looks like a very, very, very faint pink, is considered a negative. That means that acidoin was not detected. And that slight color change that you see at the top is simply due to the reagents reacting with one another and not the product of acidoin. The acidoin will cause it to turn like a bright pink red on the top of the tube. So you want to know, what does a VP positive look like? What does a VP negative look like? And why? So again, you don't need to memorize which organism does which. However, if I told you these tubes were MR, methyl red tubes, you should know that this one, if this was just labeled A and this was labeled B, you should know that A is MR positive and B is MR negative. For VP, again, you're looking for that pink red. So this would be your VP positive. This would be your VP negative. You can kind of see like this pale pink or kind of copperish color there. That is not positive. That is the reagents reacting with one another. The positive is going to be this pink red on the top. And so again, for your IMViC, the first three letters, the indole, the methyl red, and the Voges Proskauer, for all three of the first test, the pink red is going to be the positive. Now, for your citrate utilization test, you want to know the purpose. The purpose is to determine whether bacteria can utilize citrate as a sole carbon source. You want to understand that the citrate agar, the slant, is a defined media. You want to know that it's a defined media, meaning that all of the ingredients that are in the media are defined. There are no ingredients that vary from batch to batch. There are no peptones, there are no yeast extract, beef extract, etc. This is defined media. You want to know, why do we add citrate to the media? Well, the citrate is our sole source of carbon. It's our substrate. And so we want to know that the citrate is there because organisms need carbon. They need to take in carbon to make all macromolecules. And so we are trying to determine if bacteria can use the citrate as the sole carbon source. Then we have the ammonium dihydrogen phosphate. You'll notice on the next slide, I abbreviate that or I condense it. Ammonium phosphate is fine. You don't have to remember the dihydrogen part. The ammonium phosphate is there because it is the source of nitrogen. And so if the bacteria can utilize the citrate as the carbon source, they will also need a source of nitrogen. And so the bacteria would then break down the ammonium phosphate and it would produce basic products, which is our readout. So again, this test is an indirect test. We are not detecting products of citrate metabolism directly, but we are detecting the products of another process that only happens if citrate is also metabolized. So again, you don't need to know that the dipotassium phosphate is the buffer. You don't need to know that we have the sodium chloride or the agar. Those ingredients are less important for you to know. You do want to know that we have bromothymol blue in there. That is the pH indicator. And again, what colors go with which pH. For this test, you again want to study your biochemical sheet. And so when you fill out your biochemical sheet for this, we have two substrates that you should record. The citrate, this is your source of carbon, and the ammonium phosphate, which is the source of nitrogen. If bacteria produce these two enzymes, the citrate permease and the citrate lyase, both enzymes are endoenzymes, they're within the cell. If bacteria possess these enzymes, then they can use citrate as their sole carbon source and they will grow. If they grow, right, they're using a carbon source. If they grow, they also need to utilize a nitrogen source. And so simultaneously, they will utilize the ammonium phosphate. You don't need to know an enzyme for this part of the process, but you should know that the ammonium phosphate would be utilized by the bacteria and it will produce the products that are detected. The products of this test are the ammonia, the NH3, and the ammonium hydroxide, the NH4OH. These are our products. And again, these are indirect products of the citrate metabolism. We're not detecting the products of citrate metabolism directly. We are using an indirect method because if bacteria utilize the citrate, they will also utilize the ammonium phosphate because they need a source of nitrogen. You should know that they need nitrogen to make proteins and to make nucleic acids. So this is why bacteria must take in a source of nitrogen. They need it to make those macromolecules. And so if they utilize the ammonium phosphate, they will produce the products, which ultimately gives us our readout. And in this case, the ammonium hydroxide and the ammonia both will raise the pH because, again, they are basic products, so they will raise the pH, and that causes the bromothymol blue to turn blue. You should know that what the positive looks like and what the negative looks like. The positive test is going to be blue because the only way that you get alkaline products is if bacteria utilize the citrate as the sole carbon source. Or another positive result would be green with growth. Because if we see growth, the only way that the bacteria grew is if they utilize the citrate. They cannot grow if they can't utilize citrate. So if we see growth, that is still considered a positive. Maybe the bacteria has not had enough time to produce enough alkaline products for it to turn blue. But if it grows, that is still considered a positive. That is also why we have to inoculate with a needle. We have to inoculate with a needle so that we don't pick up and transfer too much bacteria. Because if I transfer a lot of bacteria, it might appear as though bacteria grew on the slant, but it didn't. You just transferred a lot. So in this test, if you see growth, that is a positive. Your negative test is going to be your green with no growth. That is your negative. Because if they don't grow, that means that bacteria did not utilize the citrate as the sole carbon source. And so that's what makes this test different than, say, a phenol red broth test. Because in this test, no growth is the true negative. That's the negative in this because the only source of carbon is the citrate. If bacteria don't utilize the citrate, they also don't grow. In the case of the phenol red broth, no growth was inconclusive because in the phenol red broth, there were peptones, there were sugars, there were multiple food sources. Bacteria should have been able to use something. So in a phenol red broth test, no growth would be inconclusive, but in a citrate test, no growth is a true negative. Because again, if they can't utilize the citrate, they can't grow. Now again, in order for that to be meaningful, you also have to check that the cap was on loose, that you didn't screw the cap on too tight. Because if the cap was on too tight, right, then if air doesn't get in, the bacteria won't grow, and you would record that as a negative but it's actually not. You just didn't do the experiment correctly. So this is what you need to study for the citrate test. This one is a little more complicated because it's not a direct test. Again, there's more than one substrate in this test. There is more than one product. There is more than one enzyme, but you want to study all of those. And you want to know, what does a positive look like and why? And what does a negative look like and why? So this, again, is just showing you the different results. You don't have to memorize which one E. coli was and which one Enterobacter aerogenes is, but you should, if given tubes, be able to tell me whether or not that is a positive or a negative. And so again, blue with growth would definitely be a positive. Green with no growth would be a negative. And so you just want to review these and practice and make sure that you could look at these tubes and be able to tell me which tube is positive and which tube is negative. And so this will conclude our review of quiz 2. So let's start with reviewing, what do you need to know for the decarboxylase test? So you want to understand that when we talk about decarboxylase, that decarboxylase is the purpose to determine whether bacteria can decarboxylate lysine or ornithine, and that lysine and ornithine are an amino group. If we look at our amino group in a decarboxylase experiment, we are removing this carboxyl group. So in terms of what you need to know in terms of the media, again, for every experiment, you don't have to memorize the entire list, but you should know the key ingredients that are in the media. So for example, you should know that the media contains glucose, because the glucose needs to be there for fermentation. Bacteria will use the glucose first at a low concentration, and when they ferment the glucose, that will produce acids, and those acids need to be present in order for decarboxylase to be active. You should know that the pH indicator is bromocresol purple, and then the various colors that it will be at the different pH. You should know pyridoxal is a required coenzyme, meaning that the decarboxylase will not be active without the pyridoxal being present. And so that's why pyridoxal is added to the media. And then know that lysine and ornithine are the amino acid substrates in the tubes. So for this test, you want to know your biochemical sheets, right? And so you should know your substrate, which is going to be your lysine or your ornithine, your endoenzyme, which is your decarboxylase. You also want to know what are the 3 requirements for that enzyme. And the 3 requirements are that the pH be low, that there be pyridoxal present, and that the tube is anaerobic, hence the mineral oil on these tubes. And then you want to know that the products are going to be either cadaverine or putrescine, depending on the substrate, and carbon dioxide. You want to know that there's glucose in the media. And again, we need the glucose to be there in order to produce acids to lower the pH. Because again, this reaction is only active if the pH is lower. You want to know, what does a positive and a negative look like? And so the positive, this is the positive over here, positive is purple. And you want to know what that purple color tells you, right? And the purple color tells you that it is alkaline and that the bacteria decarboxylated the amino acid and it produced the diamine, which raised the pH. And that's why purple is positive. You want to know that the negative is the yellow. Negative is the yellow. And that's because the pH is acidic. And what that means is that bacteria metabolized the sugar only, and that produced the acids, which lowered the pH, but they did not decarboxylate the amino acids. And so the positive is purple and the negative is yellow. You want to understand that if your substrate is lysine, that your product is going to be cadaverine. And that if your substrate is going to be ornithine, that your product is going to be the putrescine. Remember that O and P are closer in the alphabet, so ornithine goes with putrescine, lysine goes with cadaverine. And in terms of the names of the enzyme, lysine decarboxylase, the substrate is lysine. Ornithine decarboxylase, the substrate is going to be ornithine. And so you want to know the substrates, the enzymes, the products. Don't forget carbon dioxide is also a product of this reaction. You don't need to memorize which organism was positive or negative, but if given a tube, you should be able to tell me whether it's positive or negative. And again, purple would be positive. That means that the amino acid was decarboxylated and it produced diamines, which raise the pH. The yellow is the negative because the pH is acidic, which means that the bacteria fermented the sugar, the glucose, but was not able to do the decarboxylation reaction. And again, in this test, I will tell you whether the substrate was lysine or ornithine. You don't have to memorize which color cap was which. I will tell you which amino acid was in the tube. Now, phenylalanine deaminase test. You want to know the purpose. So the purpose of this test is to determine whether bacteria can deaminate phenylalanine. And so that's all you need to know from this slide. So in this experiment, you would want to know that the name of the media is that it's a phenylalanine slant. And the phenylalanine slant obviously then contains phenylalanine because it's the amino acid substrate. For this slide, you want to know your biochemical sheet. And that is that your substrate is going to be your phenylalanine, and that phenylalanine is an amino acid. You want to know that the name of the enzyme is called phenylalanine deaminase, and that phenylalanine deaminase is an endoenzyme. And you want to know the products. There are two products that are produced, the ammonia and the phenylpyruvic acid. And so the ammonia is the process of when we deaminate the amino acid. Now, you want to know that you can't use a pH indicator in this test because the ammonia and the phenylpyruvic acid will cancel each other out. The acid would lower the pH, the ammonia would raise the pH. So we can't use a pH indicator. Instead, we have to use a reagent, and the reagent in this test is going to be our ferric chloride, which detects the presence of the phenylpyruvic acid and turns green. And so you want to know, what does the negative look like? What does the positive look like? So the negative in this test is going to stay yellow, which tells us that no phenylpyruvic acid was produced, whereas the positive is going to be green, which is because the phenylpyruvic acid is present and it was detected by the ferric chloride. And so this is what you would need to know for that. Again, for this, you don't have to memorize which bacteria gave you which result, but if shown a slant, you should be able to tell me whether that is the positive or the negative. And so notice that for Proteus mirabilis, we see green on the slant. That is our positive. E. coli does not have green, it's still yellow, so that would be our negative. You want to know that a difference between deamination and decarboxylation is that deamination is going to be an aerobic process. Notice that this is on a slant, whereas decarboxylation is an anaerobic process. And that's because there's mineral oil for the decarboxylase test. And so you want to know that difference between them. Now, coming to our Kirby-Bauer. This is our disc diffusion, also known as an antibiotic sensitivity test. The purpose of this test is to perform a culture and antibiotic sensitivity test on an organism. Now, I know I wasn't the one that provided this lecture for you, but these slides were covered, and during the office hours this next week, if you have questions about it, we can go over it, but I wanted to kind of walk through and talk about what are the things that you need to know from this section. So one, you want to know that this test is standardized, meaning that when this test is performed, there are several things that have to be kept constant in order for your results to be meaningful. And so the type of agar that we use is this Mueller-Hinton agar. And then you want to know, why do we use the agar at a pH of 7.2 to 7.4? So for example, that's the pH of the blood. These drugs are going to be in the blood, and therefore we want to test these antibiotics in the blood, right? We want to mimic the environment that they're in the blood. And so you just want to go through and review for each of these parts. What is the purpose of this being standardized? What does this control for? What does this mimic? And so that's what you want to study from this. In terms of your readout for Kirby-Bauer, you want to know that you should measure the zone of inhibition, which is the clear to the clear. Now, be careful. This one starts at 0 and goes across, right? But this ruler could be set up where the 2 is here and it goes to the 4. Know that what you're doing is you're recording this in millimeters. So this 1 centimeter is equal to 10 millimeters. So if I was measuring this diameter across, right, it starts at the 0 and it goes to 3. So I would record this as 30 millimeters. So make sure you're measuring in millimeters. Now, you want to understand these 3 levels of susceptibility. So that if the bacteria is sensitive, right, you're going to have a big clearing around the disc. And that tells us that a normal dose of the antibiotic would be effective. If, on the other hand, you get a readout of it being intermediate susceptibility, that means that a higher than normal dose of the antibiotic is required, right? And so the only reason you would choose a drug that has intermediate susceptibility is if bacteria are resistant to all the other drugs, right? If bacteria are resistant to everything else, but it's intermediate susceptibility to this one drug. That means that that drug has some effectiveness against the bacteria. Not enough to just give a standard dose. What that would tell you is if a physician saw that, you would know you need to give a higher than normal dose. So either the dose itself needs to be higher, maybe it's for an extended period of time, but you can't just give the standard dose and have it be effective. And so you want to know, when do you choose to use an intermediate susceptible drug? And that is, again, if there's an antibiotic that the bacteria is sensitive to, well, you're going to choose the one that's sensitive. But if there is no sensitive antibiotic, then you would have to choose the intermediate susceptible drug, and just know that you would need to use a higher than normal dose. The other level of susceptibility would be resistant. And what does it mean if bacteria is resistant? And that means that no acceptable dose of the antibiotic is effective. You can't use any dose that it would be considered safe and have it be effective. So you would not choose a drug that the bacteria is resistant to. And again, these are all relative to the Kirby-Bauer being standardized. You have to have the test be standardized for these results to be meaningful. So you want to review this question that was posed, right? True or false, all bacteria found within the clear area around the discs are dead. So review this and understand why that's true or false. And also, is there a test you could do as a follow-up to distinguish between different possibilities? Could you do a test to distinguish if bacteria are actually dead in those clear zones? You want to know that those clear zones, again, are called your zones of inhibition. You do not need to memorize this table, okay? If on the exam we ask you to use a plate and to measure, you should be able to understand how to measure that, meaning if a ruler is given to you on a picture, if that ruler is given to you on a picture, you need to be able to know what to look for. So again, every centimeter is 10 millimeters. The little lines are the millimeters. So you want to be able to look at the ruler that's on the zone of inhibition and then compare that with this table. So if you were given this table and let's say you measured the P10 disc, this is what's written on the disc, the P10. If I measured P10 and let's say my zone of inhibition was 25, notice that 25 milliliters falls between 20 and 27. So if I'm at 25, I would record that the bacteria is intermediate susceptible to this drug, right? So you just need to be able to know how to look at the plate, measure the zone of inhibition, and then use the table, right, to determine if bacteria are resistant, intermediate susceptible, or sensitive to that drug. And so again, you do not need to memorize this table. It's going to be given to you if asked a question related to this. So some questions have come up about these tables, and do you need to memorize this table or do you need to memorize the disinfectant antiseptic table? Answer is no. The idea for the disinfectant antiseptics, which is not even on quiz 4, but the disinfectant antiseptics, it was just more for you to see different types of chemicals and how effective they were. But again, we're not going to test you. I'm not going to test you on, what type of ingredient is Clorox wipes. I don't care that you know if it's a halogen or a quaternary ammonium ion. You don't have to memorize that. You don't have to memorize the results, right? So that's not the takeaway. And same thing with this table. You don't need to memorize that clindamycin was only effective against Staph aureus. I don't care that you memorize that detail. So just be aware that when you're studying this, you don't have to memorize this table. What you do want to look at for this table, though, is you'll notice that these left 3, they're all gram negative. And if you noticed in your total number of resistance that the gram negative were much more resistant than these 3 gram positives. And so you want to take away that from this, is that the gram negative is more resistant than the gram positive. And you also want to have an idea of how do you know if a drug is narrow spectrum or broad spectrum, right? And so if you see that the bacteria, if multiple different bacteria are sensitive to this drug, right? So let's say, for example, norfloxacin. Norfloxacin had sensitivity pretty much almost all the way across, except Pseudomonas. And so if we were looking and we saw sensitive to both gram positive and gram negative, then you would say that that drug is broad spectrum. If, on the other hand, the drug is only effective against gram positive, right? So like penicillin is only effective against gram positive, or clindamycin is only effective against Staph aureus. If you saw that, you would want to know that that means that that drug is narrow spectrum. So again, you don't have to memorize what drugs are broad and narrow, but if given the data or given plates and they're labeled S's and R's for you, you should be able to look and say, well, that drug, in every case, the bacteria is sensitive. So that's broad spectrum. And if you only see some of them being affected, then that's going to be narrow spectrum. And so again, don't memorize this table. You don't need to get bogged down with that detail, but you want to understand if you were looking at this table or if you were looking at the plates, what do those plates tell you? So you want to study this slide. This is going to be useful for both lecture and lab. So you might as well study this now about which organisms are the most resistant to which organisms are the least resistant and why, what makes those organisms be more or less resistant. And so go back and review this. You want to know what it means to say that a drug is a broad spectrum versus a narrow spectrum, right? And broad kills or inhibits both gram positive and gram negative. Narrow spectrum kills only a specific type, like maybe it only affects gram positive in the case of penicillin, right? And so you want to understand what those terms mean. You also want to have an idea of why do you choose a drug that is broad versus narrow? Like under what situation would you choose a broad spectrum? Basically pros and cons of each, right? And so broad spectrum, you would choose if your patient had some sort of infection that would kill them very quickly, right? If they're in septic shock and let's say they needed a drug very quickly, then giving a broad spectrum would be the way to go because it should target, you don't have to wait to find out what's causing the infection. It should target that bacteria regardless of what it is. So that would be the advantage of using a broad spectrum. If you don't know what's causing the infection and the infection puts the patient in imminent danger, then you're going to choose to use that broad spectrum. The drawback of a broad spectrum is that if you overuse a broad spectrum, you're not only getting rid of the bad bacteria, but you're getting rid of the good bacteria too. You're killing normal flora. And so you're killing the good bacteria. That can be problematic because that can lead to what's called a superinfection. And that's an infection following a previous infection to a drug that's already been used. So basically it's that the bacteria become resistant because of the use of the broad spectrum. And so broad spectrum is not the way to go if you have time to figure out what's causing the infection. Narrow spectrum, right, is going to kill or inhibit only specific types. So if you have time to figure out what's causing the infection, well, then a narrow spectrum would be the way to go, because you're not going to be getting rid of good normal flora bacteria, right? And so that would be an advantage. Plus, narrow spectrum is also more targeted. So it's going to be more effective because it's targeting only the bacteria causing the infection. It's not being utilized by multiple different bacteria. So there are uses for both. There are cases where you need a broad spectrum. There are cases when you need a narrow spectrum. Neither one is necessarily better all the time. They have different uses. Sometimes broad's better, sometimes narrow is better. And so that's what you want to take away from this. You want to understand what is drug synergy. And drug synergy is that the two drugs combined is greater than either drug alone. So what does that look like on the plate? Again, you want to look for that enhanced synergy, or this enhanced clearing between the two discs. So like on our plates, the plates that were in the PowerPoint, the plates, if you looked at the disc for trimethoprim and the sulfa drugs, you would see that you get this enhanced clearing. It's greater than either drug alone, which tells you that the two drugs display synergy. Together, combined, they're more effective than either alone. And in the case of sulfa and trimethoprim, the reason that those two drugs are synergistic is that they both target the synthesis of folic acid, and they do so by targeting two different steps. And so combined, because they're both targeting the same pathway, combined, they do a much better job of inhibiting and shutting down that pathway than either drug alone. And if bacteria can't make folic acid, they can't make nucleic acids. And if they can't make nucleic acids, they can't divide. And so you want to understand, what is drug synergy? What does it mean to say that two drugs have drug synergy? And what would that look like on a plate? So if shown a plate, you should be able to identify which drugs display synergy by looking at this enhanced clearing between them. And so now I will review the new material for the final. And so this is going to begin with the nasal swab on MSA and the coagulase test. So basically this is going to be our lab on Staphylococcus. Then we have our throat swab on blood agar, our camp test with bacitracin discs, the A discs, or optichin, the O discs, and bile esculin. Those three labs are all part of our Streptococcus labs. Then we will deal with the yeast and the filamentous fungi. So this includes our tape mounts and our slide cultures, plaque assay, helminths, urinalysis, transformation, blood smear differentials, and blood typing. And so let's review the new material. So you have a slide that looks like this, and it's comparing and contrasting Staphylococcus and Streptococcus. And so what I did was I color coded. If it's black, it's the same. If it's red, it's different. So in terms of shape, Staphylococcus, Streptococcus, they're the same. They are both cocci, which would be our spheres. In terms of gram reaction, both are going to be gram positive, but where we start to get different is in the arrangement. Staphylococcus is going to be found in clusters, right? Staphylo refers to grape-like clusters. Strepto refers to chains. Where it's found in the body is going to be different. If you're talking about Staphylococcus, it's going to be found primarily on the skin and the nasal passage. If you're talking about Streptococcus, mouth, throat, vaginal area, GI tract, etc. Catalase reaction. So you might wonder, well, why was catalase on that list of tests to know? Wasn't catalase part of exam 2? And the answer is yes, but the reason that it's there is because catalase is a rapid test, key rapid test, that can be done to differentiate between Staphylococcus and Streptococcus, meaning if you were looking at both and you wanted to quickly differentiate whether it was Staphylococcus or Streptococcus, you could run a catalase test. because if you remember, Staphylococcus is a facultative anaerobe. It grows with or without O2, and if O2 is present, it will use O2 as its final electron acceptor. And as a result, it's going to be catalase positive. On the other end of the spectrum, if we look at Streptococcus are aerotolerant anaerobes. They're strict fermenters. And so again, what that means is that they grow where O2 is present, but they don't use it. And so as a result, Streptococcus species would be catalase negative. They don't use O2 as their final electron acceptor, and therefore they don't have catalase. Remember that they would have peroxidase instead. So if we were trying to differentiate between staph or strep, we could quickly just run a catalase test. Get a glass slide, take a little bit of the bacteria, put it on the glass slide, add a drop of hydrogen peroxide. If it bubbles, is it staph or strep? So you want to think about that. If it bubbles, is it staph or strep? And the answer is if it bubbles, you're looking at Staphylococcus, because Staphylococcus is catalase positive. They're going to take the hydrogen peroxide that you added to the bacteria, and they're going to produce water and O2, which is the gas that causes the bubbles. Streptococcus is not going to produce the O2 gas, and therefore it's not going to bubble. And so the reason that the catalase test is on this quiz and on this practical is because it's a way that we can differentiate between Staphylococcus and Streptococcus. Reaction to cold or dryness. So Staphylococcus, you have to remember, is found on the skin. So it's well adapted to dryness, right? It's tolerant to temperature changes. It's tolerant to dryness because it's used to being out in the open. So we would say that Staphylococcus is resistant or not sensitive to cold or dryness. Streptococcus, on the other hand, is fastigious, which means that it has very specific growth requirements. And so if you think about the types of places where you find Streptococcus, mouth, throat, vaginal area, GI tract, those are all very moist. Streptococcus is not resistant to dryness. It needs to live in a moist environment. In terms of salt tolerance, Staphylococcus is on your skin, therefore is osmotolerant. Streptococcus, on the other hand, is not osmotolerant. And so that's a difference between them as well. And so you want to know these general characteristics of staph and strep so that you can compare between the two and know how to identify which one it is. So you also had a slide that was comparing Staph epidermidis and Staph aureus. And so you want to review this as well. So Staph epidermidis, 100% of people carry it. So everybody is going to have Staph epidermidis on their skin as part of their normal flora. In the case of Staph aureus, some people will and others won't. So it's estimated that between 25 to 30% of people carry Staph aureus. If you are a medical worker, that goes up to about 50% probability that you carry it. In terms of pathogenicity, disease-causing ability, Staph epidermidis, not pathogenic, doesn't cause disease. It can be an opportunistic pathogen, meaning if you are immunocompromised, it can cause disease, but that's not very likely. Whereas for Staph aureus, it's more likely to be pathogenic. It's not always. There are different strains of Staph aureus. Some are much more pathogenic than others. Think of MRSA. Methicillin-resistant Staph aureus is very pathogenic. It causes severe disease. But not all strains of Staph aureus behave the same, but it can be pathogenic. Staph epidermidis is part of normal flora. Again, 100% of people carry it as part of their normal flora. In terms of Staph aureus, some people can be what are called transient carriers, meaning sometimes they carry it and others they don't. So 2/3 of people might carry it sometimes, meaning sometimes they'll test positive and other times they won't. So in many cases, Staph aureus is probably not part of somebody's normal flora, but it can be. Fermenter of mannitol. Mannitol is a type of sugar. You're going to see in our MSA agar, our mannitol salt agar, Mannitol is going to be our differential ingredient. And that's because Staph aureus can ferment it. So notice it says yes, Staph aureus can ferment it. Staph epidermidis does not. And so that is the differential ingredient in MSA to know if we're looking at Staph aureus or Staph epidermidis. And then the next one is the coagulase reaction. And so we'll talk more about this in a minute, but for now I just put this on here. So Staph epidermidis, coagulase negative, Staph aureus, coagulase positive. And again, that comes down to virulence. Coagulase is what we call a virulence factor. It's going to cause disease. So because Staph aureus is pathogenic, it has coagulase. Staph epidermidis is generally not pathogenic, so it's going to be negative for coagulase. And so that's a test that we can do to confirm whether it's Staph aureus or Staph epidermidis. So you want to understand the different virulence factors that Staph has, right? And virulence again means disease causing. And so there are three main ones that we have talked about. So coagulase is going to help the bacteria form a blood clot. So for Staph aureus, about 97% of Staph aureus would test for coagulase being positive. The advantage for having coagulase, what that does for the bacteria, is that it forms a blood clot around the bacteria and it helps shelter it and protect it from the immune system. And so because, right, if it's in your blood, for example, or it's in a cut, the blood flowing to the area is going to send white blood cells to fight that foreign invader. If staph is contained within a clot, it's going to help protect it from those white blood cells. Beta hemolysin. This is an enzyme that's used to digest red blood cells, and it uses the contents for food and multiplies. So again, this is just allowing the bacteria to then use the blood cells, the red blood cells, for food. And then lastly, we have our staphylokinase. Staphylokinase is kind of, you can think of it the opposite of coagulase. Staphylokinase is used to dissolve the clot instead of form the clot, and that allows for spread to neighboring tissues. So when resources become scarce within that blood clot, then they're going to break down that blood clot, and the blood clot, when it breaks down, it's going to allow the bacteria to disseminate and to spread to other tissues, and then the bacteria can go back to forming coagulase and form a clot and hide out from the immune system. And so you want to have a general understanding of these virulence factors that staph can have. You don't, for my class, you don't have to understand or you don't have to memorize the differences between folliculitis, abscess, furuncle, carbuncle, etc. But you do want to know the types of diseases that are associated with staph. So skin type infections, right? So an infected hair follicle, for example, it could be systemic, might cause sepsis. Staph aureus can cause pneumonia, which is a buildup of fluid in the lungs, necrotizing fasciitis or flesh-eating disease, and it's also a common cause of food poisoning. And so you just want to have an idea of the types of diseases that are linked to Staph aureus. Now, when you're studying MSA, you want to know the purpose, right? And the purpose is to presumptively identify Staphylococcus aureus and select for the growth of salt-tolerant bacteria. You want to know the general ingredients. Again, the ones that really matter are the selective ingredients and the differential ingredients. So beef extract and peptones, you don't have to memorize that those are there for general growth, but you should understand in general that that's what those types of ingredients are used for. But what you do want to know is you do want to know that sodium chloride is included in the media, and it's at a high concentration. It's 7.5%. Inside the cell is approximately 0.85%. So almost 10 times higher concentration of sodium chloride is included in the media. And that's because that ingredient is selective for osmotolerant. Skin bacteria, remember, is going to be osmotolerant. So it's trying to allow for those skin type bacteria to grow, because remember, we're trying to identify Staph aureus and Staph aureus should be osmotolerant. So we have high salt, which is selective for osmotolerant organisms. Our D-mannitol, that's our sugar. It's there for fermentation to produce acids. And again, this is going to be our differential ingredient because remember from our table, Staph aureus can ferment it, Staph epidermidis does not. And so we'll be able to look at the agar to see if it's presumptively Staph aureus. And then lastly, we have our phenol red. And phenol red, we all know, is our pH indicator. It's pink when alkaline and yellow when acidic. So for studying for the quiz, you don't have to know the procedure for this, okay? You want to have a general idea, which is that you swabbed your nasal passages. That's where you would have swabbed if you had done this. And you would have rotated that on the plate, and then you would have done a standard streak plate. So you do want to know what the different colors on the agar tell you. So again, if we see growth on MSA, growth at all tells us osmotolerant. So any growth that's on the plate is going to be our osmotolerant. Again, selective ingredients would be our sodium chloride. We're selecting for osmotolerant bacteria. Differential is going to be our D-mannitol because again, Staph aureus will ferment it. Staph epidermidis does not. We have our phenol red, which is our pH indicator. So you want to understand what these different colors tell you. So if the agar turns yellow, now I'm not talking about the bacteria itself, but the agar itself is yellow, that tells us that the pH is acidic because remember that phenol red turns yellow and acidic. We get acids produced if bacteria metabolize the mannitol. And if they've metabolized the mannitol and produce acids, it's going to lower the pH, causing the phenol red to turn yellow. That is presumptive for Staph aureus. It's possible that it could be another type of skin type bacteria called Micrococcus, which is non-pathogenic. And so if you get yellow agar, you would want to follow up with a coagulase test. Because notice that the MSA is only presumptive, meaning we can say with about 95% confidence that it's likely Staph aureus. But to confirm it, we would do coagulase. The coagulase would be our confirmatory test. Because remember, coagulase is a virulence factor. Staph aureus produces it, the others do not. Red or pink agar, right? So if the agar is pink, that tells us that it's an alkaline pH. Bacteria did not metabolize the mannitol, but instead used peptones as a food source. Right? And products of peptone metabolism are going to be alkaline. They're going to raise the pH and the agar is going to be pink. It is osmotolerant because it grew, but it did not ferment the mannitol. And so that is likely Staph epidermidis, right? It's likely Staph epidermidis because if you tested your nasal passage or you tested your skin, you're likely going to have Staph epidermidis there. And so those pink colonies are possibly Staph epidermidis. This would be a negative result in an MSA because, again, we're trying to presumptively identify Staph aureus. Staph aureus should be able to ferment the mannitol. If we did this and we got no growth, that would be no osmotolerant bacteria. So that, again, it's not Staph aureus. Now, as a control, if you were doing this, if I swab my nasal passage and I streak on an MSA plate and I get no growth on my plate, can I say that there's no bacteria in my nasal passage? Answer is no, right? Because the only thing that can grow on that plate is bacteria that's osmotolerant. So as a control, I could also take a swab and swab it on a nutrient agar plate. Because nutrient agar is going to allow lots of different types of bacteria to grow. There aren't selective ingredients. And so if I swab my nasal passage and I put the swab on nutrient agar and I see no growth, then there's no bacteria. But if I see no bacteria on the MSA, my only conclusion is that there's no osmotolerant bacteria. I can't say no bacteria. I just can say there's no osmotolerant bacteria. And so again, you want to have watched these videos to go through this more slowly. I'm just going through this quickly just to review and to help you know what you should know for your quiz. So you want to know, what does yellow agar tell you? What does pink agar tell you? What organisms is that presumptive? What did they use as a food source, etc. And so that's what you want to review for the MSA. So the coagulase test is to test whether bacteria produce coagulase like Staph aureus. And again, this would be our confirmatory test after doing an MSA. And so we would inoculate a tube that has rabbit plasma in it. And so rabbit plasma, it's just like we have plasma. The plasma is the liquid part of the blood, and it's what contains the clotting factors. And so in that rabbit plasma is going to be our substrate for this test, which is fibrinogen. So fibrinogen is a clotting factor. Now, if there's bacteria in the tube and the bacteria produces coagulase, this is going to be an exoenzyme because the bacteria needs to secrete the enzyme outside of its cell to produce the clot around it. And so this is why this enzyme is an exoenzyme. And so if the bacteria is present and they are coagulase positive, they're going to secrete this coagulase. They're going to produce the product, which is the fibrin clot, and that's going to be a solid. So that if I look at these tubes, these coagulase tests, if at the end, if it's solid, that's positive. That's confirmatory for Staph aureus. If it is negative, that means that it's coagulase negative and it's not going to be Staph aureus, or at least not a pathogenic Staph aureus because it lacks coagulase. And so just to kind of tell you how you did this, again, you don't need to memorize this for the test, but the idea is that when you did this, if you had done the MSA plate and you had from your nasal passage yellow colonies, meaning the agar turned yellow, then to confirm that you actually carry Staph aureus, you would take your culture, your yellow colony that you found on your MSA plate, and you would use that colony and you would inoculate a rabbit plasma tube. And then after the 48 hours, we would look at and see if it turned solid. If it turned solid, that would be confirmatory that the yellow agar, the colonies that form the yellow agar, were in fact Staph aureus. And so the coagulase would be our confirmatory test, because Staph aureus and Staph epidermidis, as well as other types of skin bacteria, will grow on MSA. But we're looking for Staph aureus specifically. So in terms of the throat culture, you don't have to memorize all the different types of plates that were shown as an example. Those were just to give you some example. But you do want to understand about blood hemolysis, the breakdown of red blood cells. And we'll talk about that in a minute. And so just a reminder for the throat cultures, you would have been swabbing the back part of your throat where your tonsils are. If you don't have tonsils, I don't have tonsils, you would just swab where the tonsils should be. And you would want to avoid this little thing that hangs down, the uvula, so that you're swabbing the back of the throat. And so let's look at types of hemolysis that you might see on a blood agar. We also talked about, and these are all going to kind of go together, and that is the CAMP test. And the CAMP test was done in conjunction with this. And the purpose of the CAMP test was to identify and confirm the presence of group B strep. So specifically, Strep agalacti. Strep agalacti, it's important to know if you're a carrier. There's a question in your question set that talks about a beta hemolytic strep, and to know why it's important to know if you are a carrier. And for that one, you're talking about Strep agalacti, because knowing that you're a carrier if you're female and you're pregnant knowing that you're a carrier of Streptococcus agallecti, you would be given antibiotics during delivery to try and prevent transmission to a newborn, a neonate, because if not, and this bacteria, or as the baby passes out the vaginal canal, if the baby ingests the Strep agallecti because it's in the woman's vaginal area, it can cause a serious infection in newborns. And so one way we can test for group B strep is by doing a CAMP test, and I'll describe this in a minute. And so there were 4 other bacteria that we were testing: Streptococcus pyogenes, which causes strep throat; Streptococcus mutans, which causes cavities; Streptococcus enterococcus, which is now renamed to be Enterococcus faecalis. Faecalis should tell you feces. It's in the GI tract. And then Streptococcus pneumoniae, which causes pneumonia. And so let's talk about this. So what you want to be aware is that when we talked about the setup for this, you have to realize that this was more, excuse me, this was more than just the CAMP test. We did multiple tests on the same plate. You take your loop and you basically you would make a lawn over about approximately half of your plate. So you would streak your unknown strep species about half of your plate, 2 broad streaks down. You leave a short gap, and then we also would streak Staph aureus on the bottom of the plate. This part here, the part where you're doing the lawn and the streaks down and the Staph aureus, that is the CAMP test. On the same plate, we added 2 other tests. These are not part of the CAMP test. So we did an optogen sensitivity and a bacitracin sensitivity, which is essentially a Kirby-Bauer. It's a disc diffusion method to see if the bacteria is sensitive to either of these 2 drugs, because those sensitivities would help us identify different Streptococcus species. So be aware, these are not part of the CAMP test. You don't want to say that you would expect to see, if it's optogen positive, that it's CAMP positive. No, it doesn't work that way. They're different tests. We just conserved by putting it all on one plate. And so just be aware of that. So the other thing that we talked about was bile esculin agar, because again, this goes back to identifying different Streptococcus species. And so the purpose of our bile esculin agar: select for the growth of Enterococcus for presumptive identification. So specifically for bile esculin, we're trying to presumptively identify Enterococcus faecalis. That's what we're presumptively identifying. So presumptive for E. faecalis specifically. You want to know the ingredients in this media. So review this slide, review these notes. You have to know these ingredients. So we have peptones. Peptones are there for general growth. We have oxgal, or our bile salts. This is used to inhibit gram-positive. If you think back to when we did our coliform testing, right, we had BGLB, which had bile salts. And if we were trying to grow coliforms are gram-negative, right? So in our BGLB broth, we had it there to inhibit gram-positive. In this test, it takes it a step further because the bile salts will inhibit gram-positive except Enterococcus. So this ingredient is selective. We have our sodium azide. Sodium azide is going to inhibit gram-negative. So again, this is selective. So notice that we have 2 selective ingredients. One inhibits gram-negative. One inhibits gram-positive except Enterococcus. And so what that tells you is that anything that grows on bile esculin agar must be an Enterococcus. It's the only thing that can grow. We have our esculin. Esculin has glucose in it, and it's a special food source. It's differential for E. faecalis, meaning other Enterococcuses can grow on bile esculin agar, but they don't metabolize esculin. E. faecalis does. Ferric citrate is the reagent that's in the media, so we don't add it after, it's actually in the media, and it turns brown with esculetin. And esculetin is a product of esculin digestion. And so what that means is that if the organism uses the esculin, if it metabolizes the esculin and it produces esculetin, that esculetin is going to interact with the ferric citrate and it's going to turn brown. And so let's look at that. So this slide you want to review. Okay, so esculin is going to be your substrate. It's your food source. Your product is going to be glucose. So that's the reason that organisms can use esculin as a food source. It produces glucose, which is used for food. And the other product is going to be the esculetin. And this esculetin is what is going to react with the ferric citrate. And it's going to react with the ferric citrate in the media, and it causes that dark brown color. So if we see growth on bile esculin agar, but it's not brown, so growth but not brown, it must be an enterococcus because it's the only type of bacteria that can grow, but it's not E. faecalis. That's what that tells us. If it's growth and it turns the agar brown, that is presumptive E. faecalis. Because again, E. faecalis will use the differential ingredient, which is the esculin. It will metabolize the esculin. It will produce esculetin. Esculetin will interact with the iron, and it causes the dark brown color. The way I remember this one, brown poop, right? E. faecalis, faecalis feces. Feces is the poop colored one. So the brown is the Enterococcus faecalis. And so that would be the growth, and brown would be positive for presumptive E. faecalis. So in terms of the blood agar, you want to know what the M protein is. And so the M protein was identified by Rebecca Lansfield, and it has antiphagocytic properties. It's like Velcro. It allows the bacteria to stick to surfaces. And when the bacteria sticks, it makes it difficult for the white blood cells to engulf it and to do phagocytosis. You want to review this, and that is, what are the types of hemolysis? So beta hemolysis, you're going to get clear zones around the colony. It almost looks like the agar is a little bit yellow. It doesn't turn the agar yellow. The yellow just is what appears as the red blood cells get broken down. And so the beta hemolysis is the complete destruction of red blood cells. This will be the case for both group A and group B strep, which we'll review in a minute. Alpha hemolysis. This is going to be the partial destruction of red blood cells. And so you get this green zone around the colonies. And that's because the red blood cells, the hemoglobin, gets broken down into something called methemoglobin. And it gives the agar this kind of greenish hint because the hemoglobin is not fully digested. And so of the 5 strep that we test, strep pneumoniae would be the one that would be alpha hemolytic. Gamma hemolysis, that's what this little symbol here, this is a lowercase Greek gamma. Gamma hemolysis is where we get no damage to the red blood cells. There's no change in the agar, so the bacteria will grow, but it does not break down the red blood cells. So there's no damage to the red blood cells, there's no change to the agar. And of the 5 bacteria that we test, strep mutans and group D strep would be our examples. So you want to review this slide, and just for all of the slides about the diseases that go with these different organisms, you want to have a general understanding of the types of diseases that they cause. And so strep pyogenes, right? Pyo refers to pus, so they're pus forming. And so the diseases that are associated with strep pyogenes would be strep throat or pharyngitis, scarlet fever. If it becomes systemic, it can lead to autoimmune diseases like rheumatic fever, glomerulonephritis, so damage to the kidneys. It's the most common cause of death of most common cause of necrotizing fasciitis or flesh-eating disease. It also can cause toxic shock syndrome. So you just want to kind of understand what diseases are associated with strep pyogenes. But the biggest thing is that the strep pyogenes is a type of group A strep. It's categorized as being group A strep, and group A strep is beta hemolytic. Strep agalacti is part of group B strep. Group B strep is also beta hemolytic. Strep agalacti may be normal flora in the GI tract, and some females, about 25% of females, are vaginal or anal carriers. And so this is the one, so knowing that you carry strep pyogenes is not as critical, right? Because only 5% of people carry strep pyogenes as part of their normal flora. And oftentimes if it's there, it might not be problematic. It's not something you necessarily need to know. However, knowing if you're a carrier of Streptococcus agalacti, this is the one that you need to know if you're a carrier. Especially and only really if you're female and you are pregnant. Because again, the group that's at risk for problems with strep agalacti would be neonates, newborns. And that's because if the woman carries strep agalacti or group B strep in the vagina, if she has that as part of her normal flora and the baby comes out during vaginal delivery, if the baby ingests this, it can lead to pneumonia, sepsis, and meningitis. And so again, knowing that the woman is a carrier of strep agalacti, you can give a prophylactic antibiotic during labor to try and prevent transmission to the baby so that we don't get those complications in the neonate. And so this is the one that you would want to know if you are a carrier of strep agalacti. Group C strep we're not going to talk about because group C strep is related to animals. And so we're going to skip over group C. Group D strep is going to be gamma hemolytic, which remember means no digestion of red blood cells. We have our Enterococcus faecalis. Again, it used to be called Streptococcus enterococcus and it was renamed. Enterococcus faecalis is part of normal flora in the gut. 100% of people carry it as part of their gut. Problem is when it gets out of the gut. So a gunshot wound, a stabbing, even something as simple as a colonoscopy, if they nick the colon and some of the bacteria spills into the peritoneal cavity, it can cause an infection. VRE, vancomycin-resistant enterococcus, a major nosocomial pathogen, meaning you go to the hospital for something else and you might leave with this type of antibiotic-resistant infection. And so again, you don't have to know all the details of this, but you do want to know that it's found normally in the gut and it becomes problematic if it gets out of the gut. And then our last two of our 5 that we would test would be our Streptococcus mutans, again, carried by 100% of people, and it's the cause of cavities. And so it's going to use sucrose, which is table sugar, to make dextran. And dextran is a large complex polysaccharide, and it causes this biofilm and plaque to form. Now, if you have a biofilm, right, so let's say you get that biofilm on your teeth from the bacteria growing there, and now you eat sugars, well, bacteria metabolize sugar and produce acids. The acids erode the enamel, and you end up with a cavity. And so this is a cause of cavities because it's bacteria found in your mouth, and it has the ability to form biofilms and to form that plaque on your teeth. Strep pneumoniae obviously causes pneumonia. It's the most common cause or most common form of pneumonia. There are other bacteria as well that cause pneumonia, but this is one of the more common. About 50% of people carry it. And so again, you just want to have an idea of what diseases these bacteria cause. So this slide, I was just kind of showing you how do we identify which bacteria is which. And so in our CAMP test, again, the purpose is to identify and confirm the presence of group B strep, which is strep agalacti. And so what you're looking for in the CAMP test, remember that you streak your Staph aureus. Staph aureus is also beta hemolytic. We have over here, remember I told you you're going to take about half of your plate and you're going to streak a lawn, and that is going to be your unknown Streptococcus species. And so if that Streptococcus species is group B strep, which is Streptococcus agallecti, this bacteria is producing a CAMP protein. This Staph aureus is producing that beta hemolysin, which is able to break down the red blood cells. And so those two combined, right, you also have a broad streak here. This bacteria is producing this CAMP protein. This bacteria is producing the beta hemolysin, and you get this enhanced synergy between that CAMP protein and the beta hemolysin, and you get this synergistic clearing, this triangular shaped clearing, which is characteristic of a CAMP test. So when we do this test, we had 5 unknowns. Those were the 5 Streptococcus species that I talked about. And number 3 is the one that is CAMP positive. If we look at the plate for number 3, we would see this triangular clearing. And that tells you that it's CAMP positive. CAMP positive would be Strep agallecti. And so that's what you're looking for. You're looking at this synergy between Staph aureus and the CAMP protein that's produced by Strep agallecti. And so you get this extra triangular clearing here. Remember I said that on the same plate, right, on the same plate, you would have had discs in this lawn area, and we were doing an additional test with it. Those discs on the same plate were not part of the CAMP test. They were used to identify the other types of bacteria. Because of the 5, only Strep agallecti would be CAMP positive. How then would you identify the other 4? Well, because we have differential tests for the other ones. So one of the things that we did was we had a optogen disc. This is an antibiotic. It's referred to as the O disc, and we're looking for optogen sensitivity. And remember that for antibiotic sensitivity, you're looking for these zones of inhibition, this clearing. If we see this clearing around the disc, that means that the bacteria is sensitive to the optogen. And of our 5, the only one that is sensitive to the optogen would be our Strep pneumoniae. It would also be alpha hemolytic. And for unknown number, that would go with number 2. So number 2 would have been our Strep pneumoniae. We also had another disc on the plate, which is a bacitracin disc. Bacitracin is also referred to as the A disc. And of our 5 organisms, the only one that is sensitive to bacitracin, meaning it would have a zone of inhibition, would be our Strep pyogenes. It would be beta hemolytic because it's a group A strep, so it's beta hemolytic, and that would be organism number 1. And so this would be organism number 1. So 1 was pyogenes, 2 was pneumoniae, 3 is agallecti, which means we still need a way to differentiate between Strep mutans and our Enterococcus faecalis. And so we need one more test. And so these are the differential tests to identify these different Streptococcus species. The CAMP test is used to identify group B strep or Strep agallecti. Optogen sensitivity is used to identify Strep pneumoniae. Bacitracin sensitivity is used to identify Strep pyogenes. So how do we differentiate between our Enterococcus faecalis and our Strep mutans? Because they're both gamma hemolytic, so we can't look at that. So we need an additional way to differentiate those. And so the answer is you look at your bile esculin agar. You would also inoculate a bile esculin agar, and if it grows and it's brown, it's going to be E. faecalis. And so in our example, number 4 was the one that was positive for E. faecalis. On the blood agar, it would look You would have gamma hemolysis. It would grow, but it would have no breakdown of red blood cells. And instead, you would have to then look at the bile esculin slant. We do it on a slant, not on a plate. And you would look at your bile esculin slant, and if it's brown, that is going to tell you that that organism is your E. faecalis. So number 4 would be your E. faecalis. So this slide is just summarizing your 5 unknown Streptococcus species. Put a star next to this, either this table or the next diagram. You must know this. You must know this. You must know this. So strep pyogenes, you want to know its Lancefield classification. That's group A strep. You need to know that it is beta hemolytic. And you need to know that the differential test to identify strep pyogenes is sensitivity to bacitracin. Strep agallecti would be your group B strep. It is beta hemolytic, and its differential test would be that it is CAMP positive. It has that triangular synergy between that strep and Staph aureus. E. faecalis is going to be our group D strep. It is going to be gamma hemolytic, which means it's not going to break down the red blood cells, and its differential test would be the bile esculin positive. I'm going to skip mutans for a minute. Streptococcus pneumoniae is a non-Lancefield, so it doesn't fall in this group A, B, C, or D strep. It's this other group. So Streptococcus pneumoniae is non-Lancefield. It is alpha hemolytic, which remember has a green hue on the blood agar, and it's sensitive to optogen. It's going to have clearing around the O disc. So for the strep mutans, how you would identify it in this test is it's a non-Lancefield. It is gamma hemolytic, so on the blood agar, it's not going to have clearing, and it's negative for everything else. So it's not sensitive to bacitracin. It would go right up to the bacitracin disc. It is not sensitive to optogen. It's going to go right up next to that optogen disc. It's not CAMP positive, and it's not bile esculin positive. So basically, it's negative to everything, which then by default would tell you that you're looking at the strep mutans. So I put this in here in 2 formats. So a table format, or my preferred, which is I am a very visual person, so I broke it down a little bit different. Alpha hemolysis, remember, is green, so I color-coded this green. If you see alpha hemolysis on a blood agar plate, the next thing you're looking for is optogen sensitivity. And if you see alpha hemolysis and you see optogen sensitivity, you're looking at strep pneumoniae. If the organism is beta hemolytic, then the next step is you have to look at, is it sensitive to bacitracin or is it CAMP positive? If it's sensitive to bacitracin, you're looking at Streptococcus pyogenes, group A strep. If it is CAMP positive, you're looking at strep agallecti, which is group B strep. If it is gamma hemolytic, so it does not break down the red blood cells, the red blood cells are still red, which is why this is color-coded red, then you would look for bile esculin test. If it is bile esculin positive, you're looking at Enterococcus faecalis, which is your group D strep. If it's gamma hemolytic and it's negative for everything else, by default, it is strep mutans, which is our non-Lancefield. And so you must know this. The purpose of doing all of these other tests was to help us identify our unknown Streptococcus species. And so what you would have done in class is we would have given you one of those 5 bacteria, and you would have had to figure out which bacteria you and your partner had. And so this would be how you would do process of elimination to figure out which organism you were given. So for these plates, you want to be able to look at the plate and to identify which Streptococcus species it is based on the result. And so if we look at this plate on the left, we can see that this plate is the one that is CAMP positive. It has this triangular zone of clearing, this synergy between the CAMP protein and the beta hemolysin that's produced by Staph aureus. And so we get this synergistic area of clearing here. And so when we see that, we should know that it's strep agallecti. In terms of Streptococcus pyogenes, you're looking for the fact that it is beta hemolytic. Notice that we have these clear zones, and then it is bacitracin insensitive, which tells us it's strep pyogenes. If we look at Streptococcus pneumoniae, it is alpha hemolytic. So notice that there is some partial digestion of red blood cells, but not completely. And then the other defining feature is that it's optogen sensitive. So notice there's this zone of inhibition around optogen, which tells us that it is strep pneumoniae. Enterococcus faecalis, what we're looking for in this plate is that it is gamma hemolytic, so no digestion of the red blood cells. It's not CAMP positive, it's not optogen sensitive, it's not bacitracin sensitive, but for E. faecalis, the defining feature is going to be our bile esculin agar. For strep mutans, for this one, it is going to be gamma hemolytic. It is also not optogen sensitive. It is not bacitracin sensitive, and it's not bile esculin positive. So the way that we differentiate E. faecalis from strep mutans is by looking at a bile esculin agar. And so if we're looking at E. faecalis, brown, remember, is going to be our positive. That tells us it is E. faecalis, because remember that on bile esculin agar, the only thing that grows is going to be Enterococcus. And then if it uses the esculin as a food, it's going to produce esculidin as a product, which interacts with the ferric citrate, and it's going to turn that dark brown color. So if we see gamma hemolysis along with a brown bile esculin agar, that is going to be E. faecalis. If it is negative for bile esculin and it's gamma hemolytic, then by default it is strep mutans. Strep mutans is going to be negative for all of the tests that we're looking at. So the last part that I want to go over, and I'm going to review this very quickly, I'm not going to go through all the details, but I just want to review what you want to study for your fungi. So you want to understand what mycology is, so the study of fungi. This is going to include your yeast, molds, and mushrooms. You want to know your characteristics, of all fungi. So review the list that I gave you about what are found in all fungi. So they're eukaryotic, right? Most are saprobes, etc. So go back and review the characteristics of fungi. You also want to be able to compare and contrast yeast and molds, right? And so yeast would be unicellular fungi, molds would be multicellular. When they grow on a plate, one of the things that's different is that the yeast look like bacteria. The colonies are very smooth, whereas the molds, the colonies have kind of a velvety or fuzzy appearance. You want to know that the molds are filamentous, right? They're thread-like. They have hyphae, which are filaments that are used for nutrient absorption. The molds reproduce using spores, either asexually or sexually, and we talk about examples of this. Yeast often reproduce by budding, which is an asymmetrical division. And so you want to compare and contrast yeast and mold, so the characteristics for each. You want to know an example of a medically important yeast and what disease that causes. So again, it causes candidiasis, which is a yeast infection. It can cause a yeast infection in the vaginal area or the mouth. And if it's in the mouth, again, it's referred to as thrush. You want to know that it can become systemic. So if it becomes systemic and goes through the blood, that can be very fatal for patients. You want to know which group of individuals are susceptible for Candida albicans infections. Again, some people you'll hear called Candida albicans. It totally depends on who you hear talk about it. But you want to know which group of patients are most at risk for this type of yeast infection. You want to know an example of a non-pathogenic yeast. And your non-pathogenic yeast would be Saccharomyces cerevisiae, which is your baker's or brewer's yeast. Its name means sugar fungus that makes beer. And so this organism does fermentation to produce carbon dioxide gas and ethanol. And so that's alcohol. You want to know that if you were to look under the microscope and see something like this. This is going to be the yeast, not the mold. And again, when Saccharomyces cerevisiae is growing on a plate, it's going to have an appearance that resembles bacteria. It's very smooth and it looks like bacteria as opposed to molds, which are more fuzzy. This is just another example of looking at Saccharomyces cerevisiae. Again, you can see it's these individual unicellular organisms. That's going to be different than the structures you see for molds. And so this would be your example of a yeast. You want to know the 3 types of molds that we talk about. So we have our Rhizopus stolonifer, which is our black bread mold. You want to know that their hyphae are asepty, which means that there's no septum, and that the hyphae end up being what's called coencetic. They have multinucleated cells, so there's no wall separating the nuclei. You want to know what hyphae and rhizoids are used for. You want to know that in the case of Rhizopus, the aerial hyphae is referred to as a sporangiophore. Phore tells you aerial hyphae. And it's called a sporangiophore because the type of spores that are on it are called sporangiospores, which you're going to see in a minute. So you want to know what types of spores that Rhizopus uses. So it's going to use, the structure here is called the sporangium. And within the sporangium are a type of spores that are called sporangiospores. Those are used for asexual reproduction. And you know that because there's only one hyphae going to the structure. There's no partner. On the flip side, if you see a fungal spore structure like this, this is called a zygospore. And the zygospore is used for sexual reproduction. You can see 2 hyphae coming together. We have this positive strain and this negative strain coming together, and they're reproducing sexually. So you want to know the names of these 2 types of spores, spore structures, and then which one is sexual and which one is asexual. Penicillium notatum. Okay, Penicillium notatum. This is your medically important mold, right? The big thing about this one is that it's used to make penicillin antibiotic. It's also used to make blue cheese and brie cheese. You want to know that this organism would have a septi hyphae. So again, it has the septum that separate the nuclei. It has its aerial hyphae is called the conidiophore. Phore again tells you aerial hyphae. It has these phyllides, which are these structures that hold these spores. These spores are not encapsulated like Rhizopus, so these are naked spores, and they're referred to as conidiospores. Conidiospores are again used for asexual reproduction. Notice it's coming up from one hyphae. There's no partner involved. So these would be asexual reproductive spores as well. So you want to know that they're called conidiospores and that they're used for asexual reproduction. You want to know why fungi are grown on Sauerbrood's dextrose agar. And that is because the low pH of between 5 and 6 encourages fungi to grow and not bacteria. In addition to knowing what penicillium looks like under the microscope, you should also be able to identify it by plate. And by plate, you're going to see it being the spores are going to be green and the hyphae are going to be white. And so this is what Penicillium notatum would look like growing on a plate. You want to know Aspergillus niger. So this is a medically important black mold. It causes a lung infection, which is referred to as aspergillosis. It's used to make soy sauce and soy paste. It has septi hyphae. It has a conidiophore and conidiospores, just like penicillium. So these are naked spores. Ours look more like this. So you can see this ball structure. Again, it kind of looks like Aspergillus, but the difference, it looks like Rhizopus. The difference, though, is that Rhizopus is going to be smooth because they're encapsulated, whereas these spores are naked. Again, they're asexual. There's only one hyphae here. They're asexual, and they're kinetiospores because they are naked spores. Growing on a plate, you would need to identify. So black, so think of Niger, means black. It has black spores and yellow hyphae. And so that's how you identify that. You don't, for this, have to memorize how to do the fungal plates. So you don't have to memorize that. And so again, you want to just review the pictures of the fungi and be able to identify them by plate and under the microscope. So this is what Rhizopus would look like, the black bread mold on a plate. Here is it on a slide culture. Again, the key distinction here is that the spore structure, the sporangium, is smooth because the spores are encapsulated. These are sporangiospores. They're used for asexual reproduction. So again, this is a tape mount. So not a slide culture now, but a tape mount looking at Rhizopus. And again, you can see that the spores are encapsulated, right? And so we have our sporangiospores inside this capsule structure. If we look at this one, these are going to be your zygospores, because here's our structure with our zygospore, and it has 2 hyphae coming in, which is used for sexual reproduction. If we move on to Aspergillus, you need to identify it on the plate. Again, Aspergillus niger, black are the spores, yellow are the hyphae. You want to identify it on the slide cultures. So again, they're like little balls, but the spores are naked. They use kinetiospores asexually. Here it is for a tape mount. Same thing. You can see the naked spores in this case, the kinetiospores. Again, we have 1 hyphae coming to it, so it's asexual. And then we have our penicillium. And so again, it's going to be green spores and white hyphae, so you need to identify it on the plate. These plates are easy to identify. They look very different, so it should not be hard to mix those up. Penicillium are the little fingers. They have the kinetiospores, the naked spores that are coming up. Here it is as a tape mount, same thing. So here are the phyllides, and then these are the kinetiospores. And so this is what penicillium would look like for a tape mount. So again, for these fungi, you should identify them by their plate. You should be able to identify them, what they look like in their tape mount, as well as what they look like in their slide culture. And so those are the things that you need to know for the fungi. So now let's review what you need to know for the plaque assay. So for your plaque assay, you should be able to calculate your serial dilutions. So both your dilution factors, so the amount that you're diluting each time, as well as your final dilution factors. So your final dilution factors in these tubes, as well as the final dilution factors that are on these plates. In terms of the technique for the plaque assay, you don't need to memorize the entire technique. However, you should understand that when we're doing the plaque assay, we can't just plate the virus, right? Our virus is our phage stock, our T4 phage. So you should know that that's the name of our virus, is a phage. And that when we go to plate our phage, we can't just plate it on agar alone. Because remember that viruses are obligate intracellular parasites. They can only replicate within a host cell. So what we have to do is that we need to make our serial dilution of our phage stock, and then we also need to mix that with E. coli, and we are using this E. coli strain B, which is more susceptible to infection by this phage. And so in order for this experiment to work, that we actually need to mix both together and then to pour it on the plate. Other than that, you don't need to understand the procedure and how this was all set up. So you don't need to memorize what materials are needed for this. So for your plaque assay, the really big important thing that you need to know is the purpose. So the purpose of that experiment is to determine the titer, or the PFUs per milliliter, of the original phage stock solution. You want to understand what a clear zone is. And so a clear zone is an area of cell death known as a plaque. And that clear zone is because the virus has infected the bacteria and it's killed the bacteria and caused the bacteria to lyse. And so that clear zone is a region where the bacteria is no longer living. the phage had infected that virus, or the phage had infected that bacteria and then caused that bacteria to lyse, and that's what resulted in those clear zones, or these plaques on our plate. You want to know that the strain B E. coli was used because it's very sensitive to phage infection. This slide is just showing you, again, what is a plaque. So again, a plaque is a region of clearing where the bacteria has died. And so that is due to the life cycle of the phage, where the phage is going to infect that bacterial cell and then causes that cell to rupture. And then the phage can infect neighboring cells, and then these cells will become infected and they will then lyse, and you end up with these plaques or these clear zones. And so you just want to understand, what is a plaque? Well, a plaque is a clear zone that's caused by the lysis of bacteria because that virus has infected that bacteria and caused the bacteria to rupture. For this slide, you want to know what PFU stands for. PFU stands for plaque forming unit. This is the number of clear zones that develop on the plate, so the number of plaques. You want to know that when calculating the viral titer, that you need to use the plate that has between 30 and 300 plaques. Again, this is very similar to when we did our standard plate count, but instead of counting plaques, we were counting colonies. Same idea here though. Pick the plate that has between 30 and 300 plaques. You want to know how to calculate your titer. And your titer is going to be your PFU, so your plaque forming units, divided by your final dilution factor. And so you want to know that your units will be PFUs per milliliter. So this is just an example calculation, and if given plates, you should be able to calculate titer by using this type of calculation. And so this is just an example of that. So for this, you would want to be able to look at these plates. And when looking at these plates, you would want to know which plate you should choose to calculate your titer. And then you should be able to calculate your titer based on this picture. So next we have our parasitic helminths. So when you're studying your parasitic helminths, you want to understand the general characteristics of helminths, which are that helminths are going to be worms. And these worms, because they're animals, they're heterotrophic. They're other feeders, meaning they have to consume their food. You want to know that the worms can be what we call dioecious, which means that the male reproductive organs are in one individual and the female reproductive organs are in another individual. So meaning there are separate male and females. Di means two. Some worms can be monoecious or hermaphroditic, and in that case, the animal has both male and female reproductive organs within the same organism. Mono means one. So one organism has both male and female parts. And so you want to know the general characteristics of your helminths. You want to know the types of classification of these helminths. So if we're talking about platyhelminthes, these are our flatworms. They fall into two main categories. We have our cestodes, which are going to be our flat and segmented worms. And we're going to have our trematodes, which are going to be our flat and unsegmented worms. You want to know that our nematodes are going to be our roundworms. And so of the worms that you're studying, you want to know for each worm, are they a cestode, a trematode, or a nematode? So the main thing that you want to study for your helminths is you want to study the table that you were told to fill out. And so for each of these organisms, you want to know the disease that it causes. You want to know whether it's a cestode, a trematode, or a nematode. You want to know if it's monoecious or dioecious. These are the stages that you should be able to identify on the exam. You want to be able to know what is the infectious stage and the mode of transmission. How do you get this organism? You want to know the approximate size of the adult worms. Now, you don't need to memorize size, but the purpose of the approximate size of the worm is you want to have an idea of scale. So like Enterobius, which is the one that's pinworm, that's about the size of a grain of rice. Those are fairly small worms. If we look at Ascaris, for example, those are quite a bit larger. In the lab, you would actually see these in a bottle. Taenia is very, very large, right? And so again, you don't have to memorize these exact numbers, but you want to have an idea of scale. You want to know what are the symptoms. So what are the symptoms of this type of infection? What is the treatment? And then diagnosis and additional notes. And so basically, it's the things that were written on the slides or typed on the slides. Those are going to be the important things that you want to know. So when you're studying these slides, you really want to look at what is written on the slide. Those things should be in your table, and therefore those things should be what you study. So when you're studying your helminths, you basically want to study what is on the slide. If it's written on the slide, underlined, or otherwise typed on the slide, that probably indicates that that is something that you need to know. These are the things that should be in your table. So for example, for anaerobias, you need to include in your table that this is a nematode. You want to know that the disease that it causes is referred to as anaerobiasis, or what we call pinworm. The symptoms, so here are symptoms. So many people are asymptomatic, or the main symptom would be an itchy rectum. These are the treatments. So you want to know these treatments. You want to know that this is the most common worm infection in the US. Not the most common in the world, but this is the most common in the US. You want to know that the egg is the infectious stage. And you want to be able to identify this egg. And what you're looking for is that it's oval shaped, but it does have one side that is flattened. Notice this side is round, this side is flat. And so you want to know its mode of transmission. So the way that you get this is by swallowing or inhaling the eggs through contaminated clothing, bedding, or fingers. So this is how this is transmitted, right? You have to ingest or inhale the eggs. You want to know that this worm is dioecious. There are separate males and females. Just a general thing, you don't need to memorize this detail, but just so you're aware, the male is going to be the smaller of the two. You want to know that the male has this structure called a spicule, and the spicule is a reproductive structure. The female is going to be the larger one. Again, it's about the size of a grain of rice. And you want to know that the adult worms are going to develop in the intestine. You want to understand also that one of the ways that this is transmitted is because the adult worms, the female, she is going to crawl out of the rectum at night, and she is going to lay eggs on the perianal region. And when she does that, and then let's say a child, for example, scratches and gets those eggs on their hands or in their bedding, etc., that's how this disease is transmitted. You want to know the diagnosis, so that you're going to use a cellophane tape test looking for eggs from the perianal region under the microscope. And so basically a good rule of thumb when you're studying for your helminths, if it's on the slide, you need to know it. If it's not, and I mention it, it doesn't necessarily need to be known. It's the things that are actually on the slide are what you need to study. And then you want to study your images. And so these are some of the images of these different worms. So again, this is your Anaerobias vermicularis. This is the female. So notice she doesn't have the spicule. She doesn't have that little curved structure. This is the female. You want to be able to recognize the eggs. So these are the slides that you really want to use when you're studying. These are the images that you want to know. And so this is the egg. And so notice I pointed out that one of the ways that you can identify it is that it has this one flat side. So you want to study Ascaris lumbricoides. I'm not going to go through all of this because it is in the original video, but you want to go through and you want to study this slide again. This one, remember, has two diseases that it causes. It causes ascariasis, which is going to be the adult worms in the intestine, and it also causes this Ascaris pneumonia when we get the larval worms that are in the lungs. And so basically this slide is going to be what you want to study for Ascaris, and you're going to fill in your table. So this is just showing you and comparing the male versus female. You should be able to, if you look at a picture, you should be able to identify which one is male and which one is female. And again, what you're looking for is that the male is going to have this spicule. The female does not. So that's how we can identify whether this is male or female. The Ascaris lumbricoides egg, you're looking for these mammillations, this rough edge around the egg. And so that's how you know that this is Ascaris lumbricoides. In terms of the Necator americanus or Necator americanus, however you want to pronounce this, again, what's written on the slide, those are the things that you need to study. So study this from your table. So this is going to be the images that you're going to use to study for the hookworm. This is just a different species that we used, but the Necator americanus would look the same. You can see that this is the head, and it has these little hooks, which shows us that this is the hookworm. This is just showing you the posterior or the end part, the other end of the hookworm. This is the filariform larva. This is the one that is infective to humans. And so this is the one that's going to burrow its way through the skin. And then the hookworm egg is going to have this kind of clear jelly coat. And that is how you are going to identify that one. Then we have our Schistosoma mansoni. And so again, these are the things that you want to study. You want to make sure to identify which is the definitive host. So humans being the definitive host, meaning that's where the sexual part of the life cycle occurs, while these snails are the intermediate host. This is going to be the asexual part of the life cycle. So make sure you know the distinction between the definitive host and the intermediate host. This one has two different parts to its life cycle. The definitive hosts, so humans, are where males and females are going to come together and mate. So you want to be able to distinguish between the male and the female. And so the male is going to be the one that has this gynecophorical groove where the female is going to sit in and then mate. And so you need to be able to distinguish between them. And so these are going to be the things that are written on these slides or underlined. Those are the things that you want to know for this particular helminth. So this is your picture of the Schistosoma mansoni male. So again, it has this gynecophorical groove, which is where the female is going to sit to mate. So you have to be able to distinguish between the male and the female. The female, notice you don't see that gynecophorical groove, and that's how you know that this is the female. You want to be able to identify the cercaria. This is the one that is infectious to humans. If you are swimming in water where these cercaria are found, they are going to penetrate the skin. And the way that we identify these is this mermaid-looking tail, this forked tail. You want to be able to identify the schistosoma eggs. They have this little fin on them. So again, think of it kind of like a surfboard fin. And so this is going to be our schistosoma egg. So you need to be able to identify the eggs. For Taenia saginata or Taenia solium, saginata you want to know is the one that's found in beef, meaning that the intermediate host for that one is going to be cattle. The intermediate host for Taenia solium is going to be a pig. This is the one that's found in pork. So again, you should be able to distinguish between the definitive host and the intermediate. So again, in this one, the definitive host is going to be humans again, just like in schistosoma. The intermediate host is a separate animal. So again, in this case, cattle, if we're talking about Taenia saginata, or pigs, if we're talking about Taenia solium. You don't need to know the difference in size between the two worms. That's not critical. But you do want to know that if you see the scolex, this head, and it has these little hooklets on it, these little what look like whiskers, that is going to be Taenia solium. Taenia solium is going to have those hooklets where Taenia saginata does not. And so that's a distinction between them. And so you want to know, again, the other things that are on this slide that were pointed out in the original video. You want to know that Taenia solium specifically causes an additional disease. And it causes this additional disease, which is referred to as cysticercosis. And in cysticercosis, the way that you get it is by ingestion of the eggs. And when you ingest the eggs, you end up with the larval form of the brain in the brain, the eyes, and the muscles. And so when that larval tissue gets in the brain, for example, you're going to end up with these little larval worms inside the brain. And so you want to know that the symptoms of cysticercosis are seizures and muscle or eye damage. And so that was the video that I showed you of the woman in Phoenix who ended up with this condition by eating food that was handled by somebody who basically had the tapeworms in their gut, and therefore they transmitted the eggs. And when this person ingested them, they ended up with this condition called cysticercosis. And that is, again, specific to Taenia solium. So if we see cysticercosis, if we see hooklets, those are Taenia solium. Those are from pork. So these are the images that you want to study. So again, Taenia solium has this scolex, or the head. It has this sucking disc to attach to the epithelium in the intestine. It has these little hooklets. This is how we know that it's solium. Here we have the cysticercus, the larval stage. And again, we can see these little hooklets here. You can see them here as well. This is going to tell you that this is Taenia solium. This is the gravid proglottids. Again, our slide has a little bit of a different organism here, but the idea is the same. The gravid proglottids will look the same for the Taenia solium. So you want to study this image of the gravid proglottid. These are the eggs. You're looking for the striations, these little lines in the eggs. That's what tells you it's Taenia. And so those are the things that you want to know for your helminths. Now, moving on to your urinalysis, your Kirby biplate. You want to know the purpose. And so the purpose, there are multiple purposes to this test. So the purpose is to grow bacteria from a urine sample, determine whether bacteriuria or a urinary tract infection is present, and presumptively identify the cause. So notice that there are multiple purposes to this. So to grow bacteria from a urine sample, determine whether bacteriuria is present, and also to presumptively identify the cause. You want to know what bacteriuria is. So this is going to be abnormally high levels of bacteria in the urine. And that if a patient has bacteriuria, that is going to indicate a urinary tract infection, what we call a UTI. So when you're studying this, you also want to know the risk factors for the UTIs. So go back and review your notes for the different risk factors for the UTI. You should be able to identify those. You want to understand that 90% of all UTIs are caused by E. coli. And that's because most UTIs are caused by bacteria that are present in the gut, that are normally found in the intestine. And if that fecal matter happens to make its way into the bladder, it's going to end up causing a urinary tract infection. So you want to know for your Kirby biplate that this type of agar is referred to as chrom agar. And this chrom agar is agar growth medium that turns different colors based on differences in species-specific metabolism, meaning that you're going to end up with these different color colonies on the plate, and those different color colonies are going to tell you whether or not or what type of bacteria it is presumptively. So again, it's not conclusive, but with about 95% confidence, it's that type of bacteria. When you are studying for this slide, I also indicated on this slide the selective ingredients. There are 2 selective ingredients on each side, and you want to know what are those 2 selective ingredients. So what are those selective ingredients, and what do they select for? You don't need to memorize that the side of the plate that has 1 line, that grows gram-positive only, and the side with 2 lines grows gram-negative only. That's not critical. But what you do need to know is what are the selective ingredients on either side of the plate, and what do they select for? So you don't need to memorize the procedure for how the urinalysis is set up, but this is just here for you to see. And then we need to talk about our readout. So what you need to know from this is that you want to know that when you're looking at the urinalysis plates, that you're looking for the one colony type that is the most prevalent. And that one colony type is going to be the one that you're going to count. And so if there are less than 10 of any one colony type, less than 10, that tells us that there is less than 10,000 bacteria per milliliter. And your diagnosis then would be negative. It's negative for bacteriuria. If there's between 10 and 100, that means that there's between 10,000 and 100,000 bacteria per milliliters. That is going to be considered borderline. And then more than 100 is going to be greater than 100,000 bacteria per milliliter. And that would be positive for bacteriuria. So you want to know based on these colony numbers what the diagnosis would be: negative, borderline, or positive. You do not need to memorize these colony colors. I would give you this table if asked to identify what bacteria is on that plate. So you will not be asked to memorize, that the opaque pink colonies are Staph saprophyticus. You don't need to memorize that detail. If I ask you any questions related to it, I would give you this table to help you identify what bacteria you're looking at. And same thing for this one. So this is the other side of the plate that allows the gram-negative bacteria to grow. These are the various colony colors that you might see. And based on those colors, this is your presumptive identification. Again, you don't need to memorize that. If I ask you anything about the types of bacteria that are growing on the plate, I would give you this table. So on your Herbie biplate, you should be able to calculate your original cell density. And so your original cell density is going to be your colony forming units, right? Your colony forming units. So your number of colonies of one type on that plate divided by your loop volume, which is the same as saying CFUs times 1,000. So this would be your calculation. And so you would want to be able to calculate your original cell density. And then based on this calculation, you would want to identify whether or not the patient has a urinary tract infection, right? Greater than 105 colonies would tell you that that is going to be positive, right? So greater than 100 is going to be positive. So this would be positive for a UTI. So this is what you want to know from this slide is how do you calculate, if given the number of colonies, how do you calculate the original cell density? How do you calculate the number of bacteria that were in that urine? So these are just some example plates. And so if you were given these plates, you should be able to tell me whether or not these plates are positive, negative, or borderline. So again, you're looking for the one colony type that is the most prevalent, right? So notice that on this plate, this is the one that's the most prevalent. And without counting, I can see that that is definitely more than 100 colonies. So this plate would be positive for bacteriuria. If I look at this urine sample, notice that even without counting, I know that this colony type is the most prevalent, right? It's the most prevalent. It's more than 10 colonies, but it's clearly less than 100. So this plate would be considered borderline. And so you should be able to look at these plates and say whether they are positive, negative, or if they are borderline. So these are just some other examples. These are student urine samples. Again, if you were given these plates, you should be able to say whether it is positive, negative, or borderline just based on looking at these plates. Again, these are just some more. But again, either will tell you the exact number of colonies that are on that plate, or it will be very obvious if it is between 10 and 100, or if it's less than 10 or more than 100. So you're not going to be asked to sit there during the exam and count these number of colonies. That's not going to be asked of you. So if I want you to calculate the original cell density, I will give you colony numbers. Or if I just want you to say positive, negative, or borderline, you should be able to just look and say, okay, well, that's definitely between 10 and 100, so that has to be borderline. Or that's definitely more than 100, that has to be positive, etc. And so that's what you want to know for your urinalysis. Again, you do not need to memorize what these colors represent. If I ask you about those bacteria, I would give you a table for you to compare against. So you don't need to memorize what these different colors tell you. So now moving on to our transformation lab. So what you want to know for your transformation lab is the purpose. And so the purpose of this experiment is to transform ampicillin-sensitive E. coli, so a strain that's called DH5-alpha, into an ampicillin-resistant strain with the uptake of a plasmid. You don't need to memorize that the name of the plasmid is PBr322. That's not important, but you just want to understand the purpose of a transformation. You want to know, what is a transformation? Well, a transformation is where the organism is going to take up naked DNA from its surroundings. And so in lecture, we talked about how we have a dead donor, and that dead donor releases its DNA, and then bacteria will take it up. In this case, we are artificially giving it the DNA to take up, and we're giving it by giving it a plasmid. So you want to know that during a transformation, there are a couple things that have to happen. One is that the DNA is taken up. The DNA must be maintained or replicated so that it stays within the cell, and the organism must survive the process. You want to know what a plasmid is. A plasmid is a small circular DNA that's not part of an organism's chromosome. It's an extra chromosomal piece of DNA. You want to know the general parts of a plasmid. Again, you don't have to get too specific here, but you do want to know that a plasmid needs to have an origin of replication. And so in this case, this is our origin of replication. It's like the name suggests, it's where replication begins. It's what allows that plasmid to be replicated. Plasmids have to have a selectable marker, meaning a way to select for bacteria that have our plasmid. In our experiment, our selectable marker is our ampicillin resistance. On this plasmid, there's a gene for ampicillin resistance. And so we're taking E. coli, which is normally sensitive to ampicillin, and when we transform it with this plasmid, the E. coli becomes ampicillin resistant because it acquires that gene, and that bacteria will now be able to grow in the presence of ampicillin. And so you want to understand the idea behind a selectable marker. In terms of the experiment, so what do you need to know for this experiment? You don't need to know all of the little details for how this experiment is done, but you do need to know this slide. This slide is kind of a summary of how this experiment works. And so you need to understand that the first thing we have to do is we have to make bacteria competent for transformation. And we do that by incubating with ice cold calcium chloride. And that calcium chloride, that calcium is positively charged, and that positively charged calcium is going to be attracted to the negative charge on the bacteria as well as the negative charge on the DNA. It's neutralizing those negative charges so that now the DNA and the bacteria don't repel, because those like charges would normally repel. But if we treat with calcium chloride, we're neutralizing those charges, and that allows the bacteria to be competent or more likely to take up that plasmid. Then to get the bacteria to actually take up the plasmid, we do a step that's called a heat shock. And so we take the bacteria and the plasmid that have both been in calcium chloride, we mix them together, and we raise the temperature to 42 degrees Celsius. That high temperature increases molecular motion. Those molecules start moving. We get these adhesion zones, these pores that then occur in the bacteria, which allow the plasmid to get in. Once we heat shock and allow the bacteria to take up the DNA, then we need to select for bacteria that have our plasmid. And the way that we do is we take those samples and we put it on a plate that has LB being Luria broth. It's very nutrient rich with ampicillin. And so only bacteria that have our plasmid will be resistant to the ampicillin and will grow. And so this allows us to select for bacteria that have our plasmid. And so you want to understand the overview of this experiment, but again, you don't have to know the teeny tiny little details. So again, this slide, you're not going to be asked to know. You don't need to know the steps in the procedure. That's not important. You need to know the overview, but not the individual steps. So this slide you don't need to memorize. This slide you don't need to memorize. These are just how we did the experiment. In terms of important things to remember, you don't have to know this for the exam, but you should understand general things like, aseptic technique. Obviously, we don't want to keep our tubes open for long periods of time, etc. Now, for transformation, the big important thing for this part is going to be to understand how this experiment works. So again, we're starting with our bacteria that is sensitive to ampicillin. We are making the bacteria competent to take up the plasmid. And again, we do that by treating with calcium chloride. We mix with the plasmid. We do our heat shock step that allows the plasmid to get into the bacteria. And then we take that transformed bacteria and now we plate it on a plate that has ampicillin. Only bacteria that have our plasmid will be able to grow on the plate that has the ampicillin. This slide you want to know. This is a very, very important slide. You want to know for each of these 4 plates, do you expect to see growth? Why or why not? Is it only transformed bacteria that will grow? Is it all bacteria that grows? Is there no growth? You also want to know what are some reasons why you might get a result that is different than what you expect. Meaning that for this one, like control number 2, notice what's missing from this tube is going to be the plasmid. We expect that we get no growth, right? And that's because the plasmid is what has the ampicillin resistance. If we get growth on this plate, I talked about some of the reasons why you might get that. So for example, maybe your ampicillin is old and it's no longer effective. It's no longer inhibiting the growth. That's one reason. It's possible that the bacteria naturally become resistant, right? This happens in nature all the time. So it could be that you get spontaneous resistance to ampicillin. It could be that you accidentally cross-contaminated and you actually did put plasmid in that tube, etc. So you want to go back and in the video I reviewed with you, what are some reasons why you might get a result that is different than what is expected? And so those would be the things that you would also want to study. So what do you predict you would get based on what's in those tubes? And then what are some reasons why you might get a different result? So these are just some examples of these transformation plates. So again, the experimental transformation plate should look like this. You should have isolated colonies because on this plate, we've treated it with calcium chloride. So we made the bacteria competent. We gave them the plasmid, so they have ampicillin resistance. And then we plate it on an LB ampicillin plate so that the only thing that grows is bacteria that took up our plasmid. And so we get isolated individual colonies. In our control plate, we end up with this lawn. You can see kind of right along here, this cloudiness. This cloudiness is our lawn of bacteria. That's because for this plate that has no ampicillin, it's LB only, there is nothing to inhibit the non-transformed bacteria. So on that plate, everything grows. Transformed bacteria grow, non-transformed bacteria grow, everything grows, and that's why you end up with a lawn. Our no plasmid control, we expect no growth because if we don't have the plasmid, they shouldn't be ampicillin resistant. This one has no calcium chloride. Bacteria should not be competent and therefore won't take up the plasmid even though the plasmid is there, and therefore they don't have ampicillin resistance. So you should understand, if given what's in the tube, you should be able to predict whether or not you would see growth, why or why not. And then the last part is going to deal with the blood. So both our differential blood cell counts and our blood typing. So you want to understand the purpose of the differential blood cell count to determine the percentage of various white blood cells, or what we call leukocytes, in a blood sample. You want to know that there are two groups of leukocytes. We have our granulocytes, which have our prominent cytoplasmic granules. A good way to remember that is if you see "fill" in the name, it's filled with granules. So neutrophils, eosinophils, basophils, those are all granulocytes. Agranulocytes mean without granules. They lack those granules. Notice those end in "cyte," they don't end in "fill." That's how you know that they're agranulocytes. They lack those cytoplasmic granules. In terms of the percentage of these different types of white blood cells, you don't need to know the exact numbers. Again, that varies depending on what lab you're looking at, etc. But you do want to know the general or relative abundance. So neutrophils being the most prominent, followed by lymphocytes, followed by monocytes, eosinophils, and basophils. And so I told you a good way to remember that: never let monkeys eat bananas. That's the order of abundance. So if asked a question about this relative abundance, these would be the answer choices, or numbers similar to these. They don't have to be these exact numbers. It's not going to be a trick question. Whichever one is the most abundant, that's going to be for the neutrophils. Whatever range is the next most abundant, that would be for lymphocytes. So remember that this is multiple choice. There's not going to be a trick. As long as you remember the relative order, you should be able to identify which type of what type of leukocyte you're looking at, or what type of leukocyte it might be. So when you're studying your differential blood cell counts, you should be able to identify the different types of leukocytes. So for example, if we're looking at a neutrophil, notice on these slides that all of our leukocytes have the relative abundance, the diameter, so how big they are, the nucleus, what does the nucleus look like? What about the cytoplasmic granules? And what are the functions? There's a table in your question set that go with this. You want to study this table. These are the things that you want to know for each of these white blood cells. So for neutrophils, you should know that they are referred to as polymorphonuclear neutrophils, or PMNs, because they have different types of nuclei. We can have mature or segmented nuclei, where you can see these little constrictions here. Or in this case, we're looking at a band, which is an immature neutrophil, but it is still a neutrophil nonetheless. And so again, when you're studying the size is to help you to identify that type of white blood cell. And you can compare that to a red blood cell, and a red blood cell has a size of about 7.5 micrometers in size. So a neutrophil is going to be between 12 and 15. So it's almost double the size of a red blood cell. So that helps you with the identification. And one of the reasons you're going to see that is that the lymphocytes are lymphocyte little. Those are helpful to identify by the fact that they're smaller than the granulocytes. So these are the things that you want to know for each of the types of white blood cells: the abundance, the diameter, how big they are, what does the nucleus look like, does it have granules, and what are the functions. For the eosinophils, again, you're going to identify the cell. You're looking for these red cytoplasmic granules. They contain these antimicrobial chemicals and histamines. You want to know the functions, etc. Same thing for basophils. Basophils, you're looking for these dark purple granules. That's how you know that it's a basophil. You want to know the function. The lymphocytes. Lymphocytes are little. They are not much bigger than the red blood cells. That's one of the distinguishing factors between that and the granulocytes, is that lymphocytes are little. Most are about 10 micrometers in size. And if you remember, for the granulocytes, they were between 12 and 15. So these are quite a bit smaller. You want to know, what does the nucleus look like? Well, it's spherical. There's very little cytoplasm, meaning that the nucleus takes up most of the cell. You want to know the functions, etc. Monocyte, you're looking for this horseshoe-shaped nucleus. And in this case, you can still see some cytoplasm, but it has this very prominent nucleus that is shaped as a horseshoe. Monocytes are relatively large. If you compare how big this is relative to the red blood cells, you'll notice that this monocyte is quite large. You want to know that they function for phagocytosis and that monocytes give rise to macrophages. So again, all of these things on the slide are things that you want to know. This is just to help you identify some of the different types of lymphocytes. So again, eosinophils, we're looking for these dark red stained granules. Basophils have these dark purple granules. The lymphocyte, notice that the lymphocyte, these are to scale. Look how much smaller the lymphocyte is relative to the basophil. Students sometimes have a hard time telling whether these purple dots are the nucleus or whether they are granules. The thing that you want to distinguish between them is look at the size. This cell is much larger relative to the red blood cell, whereas the lymphocyte, its size is more comparable to the red blood cells. Monocyte has this horseshoe-shaped nucleus. Neutrophil has this nucleus that can be multi-lobed in this case. In terms of your flow cytometry, you don't have to know that. You don't have to understand how flow cytometry works. I just wanted to kind of go over that with you so that you could see that there is a way to automate this rather than just sitting and looking at a slide and counting the number of leukocytes that you see. So this is just to help you to start identifying the different types of white blood cells. So this is a good thing to practice to make sure that you can distinguish between these different types of white blood cells. And then these are just more images that you should use to study of examples of these different types of white blood cells. So the two arrows on this slide are pointing to the neutrophils. We are looking at basophils. So notice these basophils have these dark purple granules. Over here, this is a lymphocyte. Lymphocyte's little. Look how much smaller it is relative to the basophil. So it's good to kind of compare to help you with identification, because I know if you look at this, this almost looks very similar to this. It can be difficult to distinguish between them. So then the next thing you should look for is going to be size. Lymphocytes are going to be little. Notice how the nucleus takes up most of the cell. So that helps you to identify your lymphocyte. Your eosinophil has this two-lobe nucleus, and it has these dark red cytoplasmic granules, which is how we know that they are eosinophils. And then we have, again, our monocyte, and it has this horseshoe-shaped nuclei. So this nucleus is how you would identify that it is a monocyte. And again, notice it's quite large. Now, next you want to know for blood typing. So for blood typing, the purpose is to determine the blood type of a person. You want to know what an antigen is. You want to know, based on somebody's blood type, what does that indicate about their red blood cells? So like if you're blood type O, for example, right, the only way that you can have type O blood is if you have the two recessive alleles. You don't have to know too much about the genetics of this, but just know that to be type O, you have to have two recessive alleles. You want to know that O means that there are no none of these A or B type carbohydrates on the surface of the red blood cells. You want to know what antibodies would be in their blood. So if somebody is type O, in their blood, they're going to make anti-A antibodies and anti-B antibodies because A carbohydrates and B carbohydrates are seen as foreign. So if somebody's type O, the only blood that they can receive safely is going to be type O. They can't receive A because their anti-A antibodies are going to react. They can't receive B and they can't receive AB. So you want to know what types of antibodies are going to be in their blood, as well as what types of blood can they receive. For A carbohydrates, right? So for type A blood, you have A carbohydrates on your red blood cells. You make anti-B antibodies because B is seen as foreign. So somebody who is A can receive O and A because O is naked. There's nothing to react with. But anything that has a B, whether it's B or AB, those are seen as foreign and those can't be given to somebody who's type A blood, etc. So this table you should know. You should know that the universal donor is O negative. The universal recipient, the one that can receive every type of blood, is AB positive. So that you need to know as well. So for this slide, you want to know, what does it mean if you're a particular blood type? What does that tell you about your red blood cells? What type of antibodies would be found in their blood? And then what blood types they could receive if given a transfusion. For this slide, you want to understand what is a hemagglutination. And so this is where we get this agglutination or clumping of red blood cells. So in this one, we are now doing something different than the slide before. For this one, we're trying to determine the blood type of a particular person. So this is the blood we're trying to type. So on this blood, there are A-type carbohydrates. So if we mix it with an anti-B antibody, this anti-B antibody recognizes B carbohydrates. This blood does not have B carbohydrates. So we don't get binding, therefore we don't get agglutination, we don't get clumping. And when we see this no agglutination or there's no clumping, that tells us that the B antigens are not present. There's no B antigens on this red blood cell. If we take that same blood and now we mix it with anti-A antibodies, the anti-A antibodies will recognize these A carbohydrates and it will bind, and what we get is this clumping or this agglutination, which tells us that the A antigens are present. So if we see agglutination with anti-A, but no agglutination with anti-B, we know that this person that we're typing is blood type A. They only have the A carbohydrates. So you want to be able to look at these diagrams or these blood typing depression plates and to look for the agglutination, this clumping. And what that tells us is that wherever we see agglutination, that means that that antigen is present. So notice for A and B, so these are anti-A antibodies or anti-A serum. If we mix that with Mr. Jones' blood, we don't get agglutination. That tells us that Mr. Jones does not have the A carbohydrate. But we do see agglutination with the anti-B serum, which means that there are B carbohydrates there. So that tells us that Mr. Jones has B-type blood, because it has the B carbohydrates, but not the A. The Rh factor you should know as well. We don't see agglutination here, so that means that this is Rh negative. So you should be able to look at these depression plates and then to determine blood type. And so this slide is just kind of summarizing if somebody is blood type A and we're blood typing them, they're going to get agglutination with the A, but not with the B because they have the A carbohydrates. Somebody who's blood type B is going to get agglutination with only the anti-B serum. Somebody who is AB is going to get agglutination with both. That means that they have A carbohydrates and they have B. Or type O, you're going to get no agglutination because they don't have either A carbohydrate or B, so that's why there's nothing to react with here. And so you should be able to look at these images and then tell me the blood type of a particular person. You want to understand how the Rh factor works. So the Rh factor is what we call the D antigen. You want to understand that if somebody is Rh positive, that means that they have this clumping factor, they have this D antigen. If somebody is Rh positive, they do not produce anti-Rh antibodies because Rh is seen as self. Somebody who is Rh positive can receive Rh positive or Rh negative blood. And in order for somebody to be Rh positive, they could be positive negative, meaning one parent gave the positive allele, one parent gave the negative, or Rh positive could be both positive alleles, so each parent gave the positive allele. Rh negative is going to lack the clumping factor. It's lacking that D antigen. Somebody who is Rh negative is going to produce anti-Rh antibodies because Rh is seen as foreign. Somebody who is Rh negative can only receive Rh negative only blood. So that's why O negative is the universal donor. Everybody can receive O negative. AB positive is the universal recipient because somebody who is Rh negative can only receive negative. Somebody who's positive can receive positive or negative blood. The only way to be Rh negative is for both alleles, both genes, to be negative negative. That's how somebody becomes Rh negative. You want to understand about Rh incompatibility. And so in this example, I gave an example of this Rh incompatibility for a female who is pregnant. And that is if the mother is Rh negative, right? If mother is Rh negative, and let's say father is Rh positive, it's possible that baby could be Rh positive as well. And if baby is Rh positive and mother is Rh negative, that's where the problem is, because when this mother goes to deliver and the baby's blood and the mother's blood commingle, well, mom's immune system is going to recognize that Rh positive as being foreign, and she's going to produce these anti-Rh antibodies. So if she subsequently gets pregnant again with a baby that is, again, a second baby is now Rh positive and she has those antibodies, well, those antibodies are going to go to the baby and they're going to start to break down the red blood cells of the baby. And so this is our Rh incompatibility. You want to understand that one of the things to treat this is that during delivery, mother is going to be given an injection of something called RhoGAM. Basically, it's injecting in these anti-Rh antibodies. And by injecting in these anti-Rh antibodies, it's preventing mom from making Rh antibodies. And if mom doesn't make the antibodies, then mom's immune system is not going to remember that, and subsequent pregnancies will be fine. So this RhoGAM is going to be critical to prevent mom's body from making these anti-Rh antibodies. And so if she's given this RhoGAM, that's going to protect future fetuses from being attacked by her own immune system. And so you want to understand about the Rh factor. You, again, should be able to look at these different blood typing samples, and any blood typing that I give you, not just necessarily these ones, but any result that I give you, you should be able to tell me blood typing. So we get agglutination for B only, no agglutination for A, no agglutination for the Rh factor. So there's no D antigen, there's no A antigen. So this person, Mr. Jones, is going to be B negative. Mr. Smith gets agglutination for A and the Rh factor, which means Mr. Smith is A positive. If we look at Mr. Green, we get agglutination for all three. You can see this cloudiness in all three. Has the A antigen, has the B antigen, and has the D antigen. So this blood type is AB positive. Ms. Brown is negative for everything. She doesn't have an A antigen. She doesn't have a B antigen. She doesn't have the Rh or D antigen. So Mrs. Brown is O negative. And so for this part, if given a plate like this, you should be able to tell me based on these results, what is the blood type of this individual? And so this is how you would go about this. And so this concludes your final review video.